Hello， 我们现场所有的长官以及贵宾，大家早安，大家好，欢迎来到2024 Nbrp 全国生医转译选拔媒合会的现场，非常欢迎我们在座所有的来宾朋友和我们一起共襄盛举。今天的议程会在3分钟之后正式开始，邀请我们已经入场的来宾，请找好座位，尽速入席。还在场外的来宾，请帮我把握时间入场。那如果在比较后方的贵宾，如果可以的话，欢迎您往前集中入座，因为在议程当中，我们会和全场的嘉宾朋友一起拍一张大合照，希望大家可以坐得稍微紧密一些些。所以后方的来宾朋友，现在请您帮忙移动位置，稍微往前，我们座位稍微集中一些些。谢谢大家的配合。议程即将在3分钟之后正式的开始，再请各位来宾于位置上稍后，谢谢。各位现场的来宾，接下来的议程会在1分钟之后开始，所以也请各位来宾尽速入席，也请各位麻烦可以移动座位，更集中一些些。待会我们会拍全场的大合照，希望我们在座所有出席的来宾都可以入镜。所以在后方的来宾朋友，以及在比较侧边的两侧的来宾朋友，麻烦帮我往中间集中，有空位都欢迎大家可以集中入席。议程马上开始，请各位来宾朋友尽速入席，谢谢大家。 各位现场的贵宾以及来宾朋友，我们议程马上就要开始了。再请各位贵宾注意，我们今天现场会捕捉大合照，希望各位贵宾可以坐的稍微再集中一些，尤其是中间的宾客，希望我们中央的椅子不要有太多的空缺，再麻烦大家配合两侧的贵宾可以来到正中央的座位来入席，谢谢大家。各位来宾，大家早安，大家好，欢迎在座所有的长官，还有我们所有的来宾，和我们一起参与第2届 Nbrp Pitch Day 全国生医转译选拔媒合会的现场。我是今天的主持人，我是 Penny， 很开心可以和大家一同共襄盛举。今天我们现场呢，非常荣幸的邀请到了来自国内外的生医创新领域的精英们。齐聚一堂，共同见证我们生医转译团队的创新成果以及合作的潜力。而本届活动呢，是由中央研究院生医转译研究中心、国家卫生研究院以及中国医药大学共同主办，并有27所的大专院校以及医疗机构参与协办，展现了跨领域的合作还有强大的力量。而我们今天呢，在议程当中呢，我们也有来自全国60多支的团队报名角逐参赛，最终我们会选出6支杰出团队以及6支潜力团队，共12支的优质队伍，给予呢奖项的肯定，希望呢可以为大家展示最具商品化潜力的创新技术。而我们活动中呢，也有安排了多场的专题演讲以及趋势论坛，相信内容绝对会非常丰富，就请我们现场的朋友拭目以待。而今天在我们活动一开始呢，我也要代表我们现场所有的来宾，一起来欢迎我们大家长来自中央研究院廖俊志院长为我们做开场致辞，掌声欢迎廖院长。
Dr. Dipinho 的各位校长、各位来宾各位同仁、各位生医界的伙伴，大家早安，大家好。今天我们第2次举办这个 Pitch Day 这个 Pitch Day 呢是这个中央研究院生医转译中心吴主任跟同仁共同的发想，不但是只有中研院自己做，而且是结合了很多单位。大家看到这个上面这个协办单位、共同主办单位有很多大学by the way， 也中国医药大学、国家卫生研究院也共同主办，然后有这么多大学，大家共襄盛举。更重要的是，我们每一次活动都吸引到数百个团队，这一次听说好像600多个团队一起来参加比赛。这个代表说是台湾最精英的前端的生医转译研究的团队全部共聚一堂，除了大家互相切磋之外，我们也请到专家来给大家做评审。那我当然知道说600多个团队，最后我们选出6个团队，很多人会没有上选，但是千万不要失望，没有选上或许是 a blessing in disguise。大部分的真正很厉害的发明，真正很厉害的研究，在早期是看不出来的，所以各位呢，今天选上当然很值得高兴，选不上千万不要气馁。那今天既然说选上选不上都没关系，为什么要来参加呢？因为这是一个很好的跟大家交流的机会，也是很好让大家能够练习怎么样 present 你自己的 idea， 能够 convince 别人你的 idea 是什么啊，然后能够得到一些 feedback， 这个我想这是这个活动的最大的意义。得不得奖倒是其次，所以不管你有没有得奖，我在这边恭喜所有参赛的团队啊，你跨出了第一步，也希望说大家再接再厉啊，把这个基础研究推到最终的目标，就是要变成救人救世的医疗用品，或者是医疗技术。谢谢大家。
非常谢谢我们廖院长为我们致辞，而且也给予我们今天全部的参赛团队一个大大的鼓励以及肯定，非常感谢您。那接下来呢，我们要邀请到的第2位贵宾哦，是前副总统陈建仁院士，但是今天院士没有办法亲自到场，所以非常的用心预录了一支影片，想要呢分享给全场所有的贵宾以及嘉宾。那么接下来就让我们一同来欣赏。
廖院长、吴主任、各位生医产业先进及转译团队们，大家好。国家生技研究园区一直以来都致力于推动国家生技医药的发展，这不仅是一项重要的使命，更是对人类健康福祉的一份承诺。透过强大的研发能力，生技研究园区成为汇集转译研究成果的重要平台，将科学创新转化为促进人类健康的实际应用。在推动生技创新方面，政府相关部会携手合作，透过精准健康战略产业的推动，使得台湾在生医聚落、生态系发展、新药与医疗器材研发制造等领域，已经取得了显著的成果。台湾生医产业市值突破兆元，这是各方面共同努力的成果，也让台湾在国际上展现了不凡的实力。经过10多年的努力，再生医疗双法在今年6月与立法院3读通过立法，在完整的法规基础上，提供了再生医学与细胞治疗相关产业更健全的发展空间。未来，台湾除了半导体与智慧机械以外，生技医疗将成为迈向国际的新亮点。融合跨领域技术的创新科技已经成为产业发展的必然趋势。政府将会持续完善产业生态系及相关法规，推动智慧生医晶片与智慧医疗产业的发展，目标是实现涵盖保健、预防、诊断、治疗、照护的全龄健康愿景，为国民健康带来更多的福祉。国家生技研究园区的 Pitch Day 是推动台湾生医转译发展的重要活动。今年的活动汇集了来自全国超过60组优秀的转译团队，所有团队将会透过竞赛展示台湾在生医领域的创新成果。我要对所有参赛的团队表达我由衷的鼓励和敬佩，你们的努力和创新精神以奠定台湾生医产业发展的坚实基础。最后，我要特别感谢中研院生医转译研究中心、国家卫生研究院、中国医药大学以及所有协办单位的努力，让这场活动得以顺利举行。我们期待大家充分利用这个机会，为台湾生医转译研究的成功发展与商业化贡献心力。祝福大家平安、健康、喜乐 ，Pitch Day 顺利圆满成功。谢谢。
感谢陈院士特别预录这一支影片，跟大家分享了未来的医疗前景，同时也给予我们今天的活动祝福，再次的感谢。那我们接着下来呢，也要邀请到呢本次的主办单位，也是中研院生医转译研究中心主任吴汉忠吴主任代表主办单位向大家说几句话，掌声欢迎吴主任。
亲爱的廖院长，来自全国各大学校长、副校长、院长， 以及我们这个相关的学研单位、跨政府单位的各部门的主管、园区跨部会进驻的好伙伴，以及我们辛苦的这个转译团队跟评审委员，以及线上现场的亲爱的贵宾，各位女士、各位先生，大家早安，大家好。The past president, MD Anderson Cancer Center, Dr. DiPino, and the past vice chancellor, the University of Texas System, Dr. Chin. Yeah, welcome visit the National Biotechnology Research Park. It is my great honor to have you join us today. 首先，我谨代表中央研究院生医转译研究中心欢迎各位莅临参加今年2024年 Nbrp Pitch Day。这一次的活动汇集了产官学研界以及医界各个重要的领域的人士参与。那我们也法人机构以及全国优秀的转译团队、业界先进、投资人一起参与了这场盛会。这一次的盛会能够如期的举行，我要特别衷心的感谢廖院长的支持，以及各大学校长、副校长、各学研机构的首长、院长们，分别担任这一次委员会这一次活动的指导委员会跟规划委员会大家一起努力来大力的支持，也让这个活动能够顺利的举办，为台湾生技产业发展贡献一份心力。Nbrp Pitch Day 规划的宗旨是要打造一个专业而且具有国际链接的生医转译合作平台。借由这个平台，我们将选拔、培育、推广及媒合来自全球的优秀转译团队，让他们的研究成果能够顺利从实验室走向市场，真正落实化研为用，造福人群。除此之外，我们也希望园区可以提供产官学研医界一个绝佳的合作及链接人脉的场域。今天的活动，我们扩大了与27个学研单位、医疗院所合作，全面选拔台湾的创新案源，强化产官学研界的合作与链接。本届 Nbrp Pitch Day 我们从各个单位推举了60对具有商业化潜力的转译团队，那也经过委员们严格的筛选，我们选出了21对入围的转译团队，再从今天在各个这个评审委员，我想最辛苦的这些评审委员，要从里面再选出6对这个获奖的杰出团队予以鼓励。获奖的团队也未来能够享受园区生医生态系的一个完善生医生态系的一个多元的价值服务。我想同时这个在与会贵宾也可以一次性全面性看到来自台湾的新创案源，并且寻找下一个生医独角兽。那我想今天还有Pitch Day 的选拔这些转译团队之外，我们也有3个重要的大会专题演讲分别来自于德州大学前校长Dr. DiPino， 还有前副校长Dr. Chin， 以及中国医药大学洪明奇洪校长。那在此OK 没关系。在此我们也有3场的这个焦点活动，其中包括 Nbrp 的学院启动，以及转译计划办公室及这个核心先导设施的启动。我想近期生技产业在带动整个国家经济，各位都知道有一个很重要的例子，在丹麦的 Novo Nordisk 的这个PLG1 这个 semaglutide 这个问世，在去年2023年销售量大增已经卖到无药可用的情况之下，他们还创造了21.2个 billion 的一个营收，也让他们的市值高达了462个 billion， 这已经超过了整个丹麦的 GDP 406个 billion。这所谓的他们的真的是国家的护国神山。 台湾的台积电也是，好，护国神山，它也 create 的这个市值已经达到了820个 billion， 好，也已经超过我们的台湾的 GDP， 好，720个 billion， 这在显示生技产业的重要性。那 Nbrp Biotrade 的成立，汇集了全国来自于学研界、产业界的顶尖人才，也为台湾生技注入源源不绝的创新能量。园区生态系已逐步完整，生医聚落也已经形成。目前园区已经有2800人进驻，包括45家新创公司厂商的进驻，37项药物已经达到临床试验，这是我们感到相当的欣慰。在这短短的时间，已经有37项的新药以及产品的开发进入临床试验，包括10家离驻的，已经有51项新药跟产品在临床试验。那也 create 2600亿的市值，如果包括离驻，已经到达3700亿的市值。我想，这个国家生技研究园区已经成为台湾生技发展的一个重要基地。所以我也要利用这个机会向转译团队们致意，你们是台湾生技产业的未来，期许在不久的将来，希望你们能够在国际舞台上发光发热，成为翻转台湾经济、推动民生福祉的最佳推手。最后，我要感谢主办单位、协办单位以及所有的工作同仁，谢谢你们辛苦的努力，让今天活动可以顺利的举办。最后在此预祝2024年 Nbrp Pitch Day 大会活动能够顺利圆满成功，也敬祝在场的所有贵宾身心灵健康快乐。谢谢大家。
非常谢谢吴主任给予我们的介绍以及致辞，请入席。就如同呢，我们主任刚刚呢在致辞当中有提到的，今天这个日子呢真的是非常非常的重要，不只是呢是我们生医转译的一个非常重要的创新力量之外呢，今天在议程当中，我们也非常荣幸可以邀请各位贵宾和我们一起见证3场的焦点仪式。在焦点仪式开始之前呢，我们现在想要和我们在座所有的贵宾先拍一张大合照，把今天这一份开心以及呢期待都记录起来。所以现在可以邀请各位摄影师以及媒体大哥移动到舞台前，各位贵宾请稍坐就可以了哦。我们在座位上来进行合照，所以侧边的朋友如果可以，现在可以再往中间集中一些些。好，那呢也请现场的媒体朋友，还有呢我们的摄影大哥可以来到舞台上哦，进行合照。好，现场的朋友，在比较后方的朋友，如果愿意，也可以稍微再往前一些些哦。那我们第1排以及第2排、第3排的长官贵宾，就帮我看一下正前方的摄影师，主要帮我看正中间这一台。那首先呢，我们先微笑的来拍一张合照哦。来，准备好，帮我看前方摄影师的镜头，请给我们一个温暖的微笑。3、2、1。好的，那我们再来拍第2张，可以请各位呢长官帮我们比一个赞吗？给我们今天所有的参赛团队，还有我们所有的生医领域的精英朋友一个大大的赞！3、2、1 ，thumbs up。很好，那我们呢再次一张，大家可以再笑得更有自信一些些哦，因为在座的真的每一位都是呢非常有这个技术水准的精英们。那我们再次一次哦，一样是赞哦，来准备，3、2、1，赞！现场的朋友大拇指都比起来。好的，谢谢大家，也谢谢我们的摄影大哥以及媒体朋友。有需要补画面的吗？好，谢谢各位。那请媒体朋友复位，也请各位的这个摄影大哥复位，谢谢大家，也谢谢所有贵宾还有来宾的配合。我们接下来呢就要进行今天非常重要的焦点仪式。今天总共有3项重要的仪式要和全场所有的朋友一起分享，也邀请你们做见证。首先呢是国家生技研究园区学院的启用仪式。 本学院呢将致力来培养具转译研究以及商业规划能力的生技领袖。那接下来呢，我就要邀请贵宾来到台上，为我们正式启用。首先，我们就欢迎中央研究院廖俊智廖院长，请上台。同时呢，我们也欢迎中国医药大学校长洪明奇院士、中国卫生研究院司徒惠康院长，以及我们接下来要欢迎来自美国的贵宾。Now we would like to invite our distinguished guest, Dr. Diping Niu. Please go on stage and have the launch ceremony with us. And next, we would like to invite Dr. Linda Qing. Please go on stage and be with us. 那接下来呢，我们要继续欢迎中研院生医转译研究中心吴汉忠主任。国科会生命科学研究发展处涂君怡副处长，以及国家实验动物中心秦贤进主任，生物技术开发中心涂醒哲董事长，以及中研院生医转译研究中心客座讲座教授柯承恩柯教授，欢迎大家，请上台。那么稍待一会儿呢，当我们倒数3、2、1的时候，要麻烦各位长官将您的右手帮我放置在前方的启动台。After I count down 3, 2, 1, please press your right hand on the in front of you, OK? OK， 好的，那我们如果准备好的话呢，我们现场的各位来宾朋友，我们就一起倒数，准备迎接我们的启用仪式正式启动。倒数3、2、1，请启动 Nbrp 学院国家生技研究园区学院正式启用。本学院呢，预祝我们生医领域所有创新的未来都可以呢前景无限。各位长官，请维持这个姿势，帮我看一下正前方摄影师，请给我们一个自信的微笑。谢谢各位长官为我们正式启用国家生技园区学院。那我们第2张可以给我们一个赞吗？好，来，一样帮我们用你的左手帮我比个赞。希望呢，我们国内生技创新的人才在商业思维以及技能都可以透过本学院呢来获得支持，创造台湾生医产业新的价值。谢谢各位长官，各位长官请帮我留在原地不动，因为这个学院可以落成，除了在座所有长官的努力之外，我们真的还有很多伙伴和我们共同完成。所以接下来我要来邀请我们的贵宾一同来到台上，为我们一起合影。我们欢迎生物技术开发中心陈秀辉代执行长，中央研究院生医转译研究中心的各位同仁，林荣信副主任、李文山副主任、陶密华执行长、杨瑞斌执行长以及施修明执行长，欢迎，请上台。接下来欢迎来自宏基制医连家恩董事长，还有呢我们洪伟仁顾问以及刘祖慧博士，欢迎，请一同上台，帮我站在呢我们的第2排的高台。那也请呢我们第2排的长官稍微帮我穿插在前方长官的肩膀的位置。好，来，我们呢稍微移动一下，对，可以。好的，来，把头都可以露出来哦，都可以清楚的看到我们的摄影师。好的，非常感谢。那我们接下来就请各位长官，我们看向摄影师，给我们一个大大的赞。谢谢大家的掌声，我相信呢，这个掌声里面也代表了非常多的期许。未来我们台湾的生医产业的价值绝对可以再创新高。再次感谢我们各位长官，我们再帮我看到正前方哦，我们再次的3、2、1，最后一张就可以让媒体的朋友可以捕捉到最完美的画面哦。准备3、2、1，赞！好的，非常感谢我们台上所有的贵宾，辛苦了，请先入席。谢谢你们为我们正式启用国家生技研究园区学院，我们也期待未来可以看到更多生医转译的团队技术，可以在呢我们的商业以及这个呢我们生医的这个人民福祉中呢来进行这个发光发热。那么 么接下来呢，我们要进行的是 Nbrp 学研转译办公室的揭牌仪式。本办公室呢是期许促进跨校以及医院的合作，提升研究的转化成果。所以我们接下来呢，还是要再次的来欢迎我们贵宾上台，为我们呢一起来做揭幕哦。我们再次的欢迎中央研究院的廖院长，请上台，以及我们中研院生医转译研究中心吴主任，请上台，请帮我站在呢我们的揭牌的看板的左右两侧哦。接着欢迎中国医药大学洪明奇校长，以及呢国家卫生研究院司徒惠康院长，中研院生医转译研究中心 AAC 召集人张宏仁董事长，以及高雄医学大学于明龙校长，台湾大学附设医院吴明贤院长，以及台北医学大学吴麦斯校长，国立东华大学徐辉明校长，以及国防医学院陈元浩校长，以及国立宜兰大学陈威荣校长。欢迎以上所有贵宾帮我来到舞台的正中央的两侧。那我们有一个红色的这个缎带，麻烦您们帮我一起握着哦。我们这一个呢揭牌仪式呢，其实也是象征着我们的中研院携手了全国27所大学，来促进医院和学校的合作，成为呢我们产学业界合作的重要枢纽。那你待会呢，也让我倒数3、2、1的同时，我们就一起拉开红布，正式揭幕。那我们大家准备好，倒数3、2、1，请揭幕！国家生技研究园区学研转译办公室正式成立，预祝我们产学合作可以顺利通畅，让技术可以发扬光大。那我们就请各位长官呢，麻烦帮我看一下正前方的摄影师，请给我一个呢自信的微笑哦。准备好，3、2、1，请帮我看一下正前方。好的，谢谢我们的所有贵宾，请帮我们先行稍后，因为我们还有第2批贵宾可以跟我们一起合影吗？我们时间应该还可以。好的。好，那我们接下来就欢迎呢我们第二阶段的贵宾一同上台哦。我们欢迎中研院生医转译研究中心的林荣信副主任，以及李文山副主任，以及国立阳明交通大学的郑子豪副校长，以及国立中兴大学陈全木副校长，一手大学陈韵如研究发展处研发长，以及长庚大学的陈继勋暨合长，以及我们中研院生医转译研究中心顾问张大慈顾问，以及台大产学合作总中心杨宗林副主任。欢迎，请帮我们站到第2排的站台上面。好，来请，那也请帮我站在第1排贵宾的肩膀跟肩膀的中间，让你们的脸都可以完整露出哦。好的，来，请上台，请上台，谢谢各位贵宾的配合。好，那我们请看一下正前方的摄影师。请准备，也请给我们一个呢非常温暖的微笑哦。来，准备，3、2、1，请看正前方。谢谢各位长官，一起促成此次的合作。感谢我们台上所有的长官，谢谢您，同时也谢谢廖院长以及我们台上所有的贵宾，请入席，谢谢，辛苦了。那我们呢还是。辛苦我们今天在场所有的贵宾哦，为我们揭晓今天第2个非常重点的仪式，也让现场所有的嘉宾知道，未来我们的技术无论是在学界、产业界都可以有顺畅的交流。那我们接下来呢，要进行我们今天第3项焦点仪式，要一起见证核酸先导设施的揭牌仪式。那我们接下来呢，要再次的欢迎廖院长帮我们来到舞台的正中央，谢谢你。同时，我们也要欢迎中央研究院的陈君宏秘书长、生医转译研究中心的吴主任，以及呢李文山副主任，以及陶密华执行长，也帮我一同来到台上。同时，我们也欢迎执行团队的林英秀博士、江博正博士、张德斌博士，以及傅彦明经理，和我们一起共同的来做揭幕。我们这一项核酸先导设施是国际级的一种设施哦，象征着台湾核酸药物临床的迈进 新高度的一个全新的设计，从设计、生产到临床实验都提供完整的服务哦，而且都具国际的标准。所以各位来宾，如果已经上台，帮我握紧我们红色的缎带，待会我们就正式的来揭牌。好，来准备现场的朋友，请倒数3、2、1，正式揭牌。核酸先导设施正式落地，希望透过这项设施呢，可以让研究团队呢有更多专业的支持，加速研发的成果。那我们也请各位贵宾看向正前方的摄影师，一样请给我们一个呢非常自信的微笑，也预祝呢我们在核酸的医界领域可以再创新高。谢谢各位贵宾。和我们一起共同见证今天3项重点的仪式，再次的感谢廖院长，也谢谢我们在座所有贵宾，请入席。真的非常荣幸可以邀请到大家和我们一起见证了我们生医领域的这个3项重点的仪式。那接着下来呢，我们会先进行中场休息，下半阶段呢，我们会展开今天第1场的专题演讲。我们下一场议程会在10点40分，也就是35分钟之后再次的开始，所以再请各位来宾帮我留意时间。那我们这一次会场在1楼会备有茶点区，同时也有这一次呢参赛的组别完整的海报介绍以及赞助商的摊位，所以再请各位来宾可以利用休息时间来到1楼来取用茶点，同时呢可以帮我们参观了解。我们待会10点40分再次回到现场，谢谢各位来宾。
this microphone?
You want to hold it, or you just?
You know what? I will, since I have the remote, I will kind of stand
Okay, so
here
just take
instead
it.
of behind the
I think you just take it. Oh, okay, use
Yeah,
this one.
I will do that. Yeah, that would be better. Perfect. Okay, good.
Okay.
Excellent. Thank you.
他会拿，我们会直播，所以他如果这样子走来走去，他会走，他在中间。
哦，那他手拿？
不是，但是你的直播的画面
会啊，
哦，
会
可以哈。
嗯。
好。
他确定要走来走去？
对，他会走来走去。
邀请我们这一场所有的长官以及来宾尽速入席。接下来的专题演讲非常的精彩，会在3分钟之后准时展开。邀请还在场位的贵宾把握时间回座，谢谢大家的配合。好了，我们现场所有的贵宾，欢迎大家再次回到今日的议程现场。我们接下来的专题演讲马上在1分钟之内就要开始了，所以请各位来宾请加紧脚步回到座位上，一起迎接我们今天第1场精彩的专题演讲。 Hello， 欢迎现场所有的贵宾以及各位所有来宾朋友，再次的回到 Pitch Day 今天的议程现场。我们接下来紧接着进入今天第一场的专题演讲。我们非常荣幸呢，可以邀请到的第一位演讲嘉宾是来自于美国国家科学院院士及美国德州大学 MD Anderson Cancer Center 前校长 Dr. Ronald DePinho， 欢迎。Dr. DePinho 今天来到现场，为我们即将来进行简报。他将带来的呢主题是生技产业将知识转化为患者福祉的主题。那在我们演讲开始之前，我们也非常的荣幸邀请到了中国医药大学的洪明奇校长来为我们担任引言人，掌声欢迎洪校长。
Okay, good morning, everyone. It is a pleasure, and actually, I feel extremely honored to be able to be here to introduce my boss, Dr. Ronald A. DePinho, that past president of MD Anderson Cancer Center. Dr. DePinho is a renowned cancer biologist in United States and in the world. He is a cancer leader. Dr. DePinho has a lot of things that we can learn, especially for this pitch day. He received his MD degree from Albert Einstein Medical College. And for those of you who have read the newspaper, that I believe it was last year, there was a big donation of $1 billion US, 300亿台币, okay, to that Albert Einstein Medical College. And then what I heard in the news is in the future, the medical student over there, tuition free. But Dr. DePinho already paid his tuition. Too late. But anyway, in the future, okay? And he received the MD degree over there. After that, he spent quite a few years, actually, I believe the 6 years in New York City, Columbia University Stadium Medical Center. Over there, he finished his fellow training and also spent 4 years full-time 7-11 laboratory research postdoc fellow training. He didn't receive a PhD, but his research skill is much better than PhD. Sorry, this is the time that it's not easy to find the time to embarrass my boss, my ex-boss. So I need to spend some time to embarrass him. And his postdoc fellow work actually already received the world attention. Therefore, he was recruited back to his mother institution as an assistant professor, and all the way spent 10 years there, very quickly ranked to the full professor, but he has done so well that Harvard Medical School over in Boston then provide him a package that he cannot refuse. So he spent quite a few years, in more than 10 years, in Boston Harvard Medical School, where he actually put a lot of effort, try to convert the academic basic research translate into the product. And as a matter of fact, if you look at his CV, he served as a founder for spin-out company, a scientific advisor, chairman of scientific advisor, at least 20 different companies. I'm not kidding, okay? So then, since he did so well, that University of Texas MD Anderson Cancer Center, where I actually served for 3 decades and retired, then come back to Taiwan. At that time, 2011, University of Texas was so lucky be able to convince Dr. DePinho to come to MD Anderson to lead MD Anderson Cancer Center. So for those of you who probably know that there's a term called 癌症登月计划 cancer moonshot, right? a lot of people in Taiwan thought the cancer moonshot was initiated by US President Obama, right? Wrong. It was initiated in 2011 when Dr. DiPino came to MD Anderson, but his vision is there's so much data, so much knowledge already available in the literature. So he always tells us a term called low-hanging fruit. I don't know whether he still uses that term. And then he initiates a cancer moonshot. And so his goal is to be able to, using the current knowledge, quickly to convert that into, to help the cancer patient. After a few years, he transformed MD Anderson Cancer Center. He then passed the MD Anderson leadership to another junior person. Then he came back to continue to lead the cancer research, not only in the United States, globally. So he actually puts a lot of collaboration internationally, and also put a lot of effort on translate from basic research outcome into spin-out company. His achievement has been admired by many, many people, including me. Then he has received tremendous number of awards. He was a member of National Academy, U.S. member of National Academy of Science, a member of National Academy of Medicine, and he was a member of the American Art and Science Society. So with that, today, I think we are very lucky to be able to invite him to come to share with us that his knowledge on the translation, in particular in this pitch day. Before I hand to him, there's something very, very important that the audience need to know. You have to be very nice to him, because he's a black belt, karate. He used to be in the U.S. Columbia, U.S. What is that? 世界那个 yeah, he was the U.S. team, black belt, karate, and he's still doing that. So be very nice to him. Okay, okay. So no. Okay, so.
Oh my God. Thank you. Well, Director Wu, thank you for the honor of being here to share some words with this august group and all of the leaders here. I admire this institution, I admire this country, and of course I admire Min-Chi Hung. Whatever Min-Chi Hung tells me to do, I just say yes. But also, thank you, my friend, and congratulations on all of your success as well. So today I'm going to tell you about a few things that we're going to talk about a lot of different things that I hope will be relevant and interesting to the diverse audience here. These are my disclosures, which kind of reflects the fact that I have had the privilege of being able to be involved in a lot of biotechnology companies. I was the main advisor for GSK and for Abbott at one point as well. So those have really given me some insights into how the ecosystem works and what are the critical issues that one needs to think about to be able to convert knowledge into things that actually matter for patients. And this is what I'll be telling you about. Why should we even bother to create a biotech ecosystem? What are the elements that are important for creating that ecosystem? And I can't resist but to tell you about research, where the field is going, but also I'm going to give you a couple of illustrative examples of what my team has done in the area of pancreas cancer, in the area of aging, just to give you a sense of how science has actually moved to a very exciting point in its history that can really make a decisive assault on pain and suffering around the world. And I was having this conversation in the VIP room just a few moments ago. You know, as Taiwan goes, as the United States goes, as Europe goes, as China goes, so goes the world. We have to work together. It is our responsibility to work together as a scientific community, as a healing community, to really help patients. There's a tremendous amount of pain and suffering around the world. It's our responsibility to change those stories. And so why create a biotech ecosystem? As much as I love science, I've often said that no Nature paper has ever cured a patient. It's only when you convert that knowledge, that insight, into a new drug, a new diagnostic, or if you're in the government, a new policy that's evidence-based, that can really make an impact on patients. And then it's important to realize that the relationship between biotech and academic institutions is a symbiotic one. 1 + 1 = 20. That is that investments in biotech are going to help the academic institutions to bring resources back in the way of licensing revenue and so on that can fuel its mission of education and patient care and research. And then reciprocally, obviously, academic institutions play a very important role as well. And then I think it's important to appreciate that really the biotech industry is one that returns not only monetary value, but societal impact. It's very important to recognize that a lot of people are counting on us. And I think I just used one example of mRNA vaccines that actually saved the world, right? It had a huge impact, and that was decades and decades of supposedly irrelevant basic science that we had no idea was going to actually be one of the critical technologies that enabled a vaccine to be developed and to really reduce the impact of COVID. And so this is also another important context, which is that there are theoretically thousands and thousands of diseases, but we only have a very small fraction of those diseases for which we have some cure or even some kind of mitigating treatment. And it's important to appreciate that by next year, there are going to be over 1 billion people around the world over age 60. I'm one of them. And so you have, after 60, you have this geometric increase in the incidence of cancer, diabetes, heart disease, and Alzheimer's, culminating in risk of cancer by age 80 to 85 of 40%, 1 in 2 men, 1 in 3 women, and then Alzheimer's, close to 40% will get dementia, and Alzheimer's is the main cause of dementia and so on. So this is a big societal impact, and with the aging of the world population, this is going to be increasingly more and more important. So we really have to have a sense of urgency to make an impact on these issues here. And so how do you create a biotech ecosystem? If you study Boston or you study the Bay Area, any of the ecosystems that are now mature, what actually had to come together to make those things happen? And there are really 3 pillars, 3 foundational elements that must work together. And if you remember anything from this talk, it's to recognize that you really need multi-sector collaboration to be able to make things happen. This country is what it is because of visionary leadership decades ago that recognized that the semiconductor industry was going to be the future oil of this world. And information was going to be very, very important. That was, in private sector, government, and academia, basic research coming together to be able to make that happen. And so I think there's a unique opportunity as we go forward for this country to use the same cultural elements to really make that kind of impact. So what does it really take? So government has a tremendous responsibility in simply funding research. There's a very direct link between national vitality and government's investment in research and innovation. It is a direct correlation. And the government can also mitigate risk by putting in support for early stage biotechnology that will incentivize the private sector to put resources in. And of course, it plays an extremely important regulatory role. Its responsibility is to protect the patient. And so you have to make sure that the drugs that are being developed and so on have the highest standards possible. And then academia, of course, plays a role in creating knowledge, right? Basic, translational, clinical research, and clinical trials infrastructure. I can't emphasize enough how important it is. If you want a robust biotechnology industry, you have to have a robust phase 1 clinical trials. These novel agents that are coming and being developed have to have a handoff into extremely skilled, highly capable phase 1 trials infrastructure. Could not emphasize this enough. And then technology infrastructure. You know, these early stage biotechnology companies, they can't invest in, you know, mouse colonies and, you know, all the sequencing and all of those kinds of things. It's just too expensive to essentially reinvent the wheel. And then, of course, our academic institutions, and this is happening here today, is to train the faculty, to train the students in entrepreneurship, in business, and in science, right? All of those things are very important. And then the private sector plays a very important role. When it thinks about investing into a company, the really sophisticated blue chip VCs conduct very robust due diligence. You think it's hard to publish and sell in Nature? Well, to be able to get past diligence, from a blue chip VC to create a company and get investments in that is 100x what it takes to get one of those publications in. And you need not just scientific excellence, but intellectual property excellence, business development excellence, regulatory excellence, so on and so forth. And they also provide operational expertise, seasoned management, and the hundreds of millions of dollars needed to be able to actually bring something all the way to a proof of concept and beyond to a new FDA or new drug NDA. Okay, so I want to emphasize with that just a couple of examples of what each of those elements have done to really make a difference. So, and I'm going to give you some just general points, which is that government's investment in academia is absolutely the most important thing. You need to have a robust academic research enterprise if you expect to have a biotechnology industry. And of course, in the United States, the examples are the NIH, the NSF, ARPA-H, which is essentially the DARPA for health that President Biden created. It was actually inspired by the MD Anderson Cancer Moonshot. And then we have early stage biotechnology investments. And here this is very important. So in Texas, we have what's called CPRIT, which is a $3 billion fund over a 10-year period. 2/3 of that goes to science, but 1/3, $1 billion, goes to, in a competitive way, give grants to new early biotechnology companies. And so I've been, helped a number of countries help their biotech ecosystem get going. It's very important for the government to put risk capital into these early stage biotechs. So for example, in Texas, if you you can get up to $20 million for a company, and you have to match that with at least $1 for every $2 of the government, state of Texas investment. So imagine you're an investor, you're only putting in $10 million, and you get another $20 million of non-dilutive capital. So that's a tremendous incentive to be able to create companies in Texas. So the government can play those kinds of roles. And then, of course, it has the small business research grants from the NIH, which are less money, but still very, very helpful. And then academic institutions have to invest in people and platforms. And I can't emphasize this enough, but you really need good technology transfer offices. The typical academic tech transfer office really needs to be upscaled so that they really understand patents, they understand business development, how to make deals so the institution does well from a return on investment standpoint. And then really the clinical trials infrastructure that I mentioned, many different technologies that really enable the companies to take flight and so on. And then educational programs, right? At the end of the day, academic institutions are about training the next generation in areas of entrepreneurship, business, management, not just science, right? So all of those components are very important. And then the private sector has to invest in the community, right? It's not just about putting money in and getting money back, but you saw this in Boston, where the real estate industry and so on all came together to create Kendall Square. Within 1 mile of Kendall Square, there's more than 1,000 biotechnology startups, right? So that is an example, and then also the VCs really help create this very seasoned management, which is really critically important, right? You think about the science and the IP as raw material. That has to be organized into value that will return value to the patients and return value to the investors. Now, one of the things that I'm often asked is, when should I start a biotechnology company? And I thought it's very important to recognize that through the lens of investors, when they put money in, they want to get money back, right? It's not a charity, it's a business. And so you have to think about, well, what is the return on investment? What is the risk at every stage along the way? And essentially, when you start a company, the optimal time to start a company is when you have a development candidate. You think you have a drug that's good enough to bring into a human being. That window, just before IND-enabling studies, right, where you do toxicology, manufacturing, formulation, all of those things, is the optimal time to start a company. The value of an asset, a development candidate, is around $25, $35 million, right? And it takes about $40 million to go from there to clinical proof of concept, which starts to happen in phase 1B or early phase 2. You don't even have to complete phase 2. You should have not only safety, but some signal of activity here. And then once you have that, that's the optimal time to go and have an exit, either an acquisition or an IPO. And the value, the average value post phase 1 is about $505 million. That's about 700 companies worth. So you go from about 25 to 500, and the amount of money that's put at risk is about $40 million. So it's pretty good, and the risk, depending if it's antibody, small molecule, is actually quite reasonable. So you can have more or less 1 in 3, 1 in 2 to 1 in 3, if the science is good and you've done your due diligence, that can make it. So it's a tremendous return, and it only takes about 5 or 6 years to go from that point to that point. Optimal entry and so on, okay? So that's just from the economics. But now let's talk a little bit about the risks. And here's where, if you set this up correctly, you can significantly mitigate the risks. So, when you start, made a discovery in the lab and you start doing drug screens, as you do here at Academic Seneca, and then you have, your drugs that you bring forward into clinical trials, the rate of attrition is tremendous, right? And so, for every drug that's entering into clinical trial, only 1 in 20 becomes an approved drug, right? And so this is significant. And most of the failures, surprisingly, actually happen in phase 3 clinical trials. And so what can you do to mitigate the risk? Right? If you were to design it, and you have the opportunity to design it right from the beginning, right? So first of all, there are scientific solutions surrounding companies with world-class science advisors, obvious, but people who really know the science and understand biotechnology. And with Zoom now, you can have world-class investigators around the world, the very best people. And so the other is to get very robust preclinical validation of the target on the genetic as well as the pharmacologic levels. Most of those failures that occur in phase 3 is because the target and the drug were not truly validated and patient selection was not really understood, right? This is so important, and I'm going to return to that point. And then you really need that strong clinical hypothesis. You'll hear more about this from Dr. Chin. And then there's operational issues, right? It's not just about the science. You actually have to get the stuff done, and you have to do it properly. and you have to get through regulatory and so on. So you really need to create startup teams with highly experienced professionals. I'm going to give you one organizational construct that BioTrek could potentially consider. And then recruiting drug developers with a record of success and integrate experts across all of these different diseases. It's not just about drug development. You need the biology and you need the clinical insight. You really have to bring those three elements together, which is what we did in the moonshot. So the reason why the moonshot was started was to integrate across drug discovery, biological insight, and clinical knowledge, right? And normally it's just a relay race, you know? You do the basic science, then you hand it off and you do the drug discovery, you hand it off, you do the clinical trials. Huge rate of failure. Bring all those elements together from the beginning, you can have a huge impact. And then obviously there's an opportunity to actually be very efficient and have legal, HR, accounting, IT, all of these things in a more holding company fashion that allows you to fractionally utilize those very strong seasoned assets. So this is the typical startup biotechnology flywheel. This is what happens thousands of times each year. People start companies, right? So you have a discovery or a platform, you create a C Corp or an LLC, you license the technology into that, you secure financing, you recruit key managers and advisors, you build your management team and scientific team, you scale operations, create value, and then you have your exit, right? Reinvent the wheel, every single company, each time. Is that the most optimal way to do it? Well, one possibility is that you have a management team that really helps facilitate asset development and helps manage different companies so that these companies can do this in a very capital efficient manner. They may be able to hire a fractional CEO or a fractional CMO that can really mitigate a tremendous amount of expense, but also bring very, very high quality individuals into the company. So what this allows one to do is to be very efficient, to have experienced on-demand management across multiple companies, shared services for portfolio companies like HR, accounting, things of that nature, and then having internal drug discovery for lead clinical candidate asset creation and so on. So this is kind of the Bridge Bio model, which proved to be very interesting. A company that I started, Sporos. This is also the model for Apricity Health, which you'll hear about tomorrow. But it's the idea that you really have a very seasoned team here that is able to cultivate assets, and then as those assets mature, you then create new companies and so on. Okay, so now let's just come to one key point, which is important for our academic colleagues in the labs to really understand. And that is that when you think about the failures of clinical development, as I mentioned, the 95% failure rate from here to here, only 1 in 20 make it. Half of those failures actually occur in phase 3, when you're spending hundreds of millions of dollars, a couple of thousand patients, right? Hundreds to thousands of patients. And so this is really a huge problem. And really the reason for this really gets down to the fact that the target was not well validated and the drug is not hitting that target specifically. And then most importantly, it's not knowing what fight to pick in the clinic. That is, you don't know which patient population to use that drug in. Now, of course, if you have EGF receptor mutations in lung cancer, you know you're going to get a response there. That's different, right? And that's an example where you have a very good idea of which patients to use which drug for, et cetera. And so it's very important, especially when you have novel agents and so on, that you really do your homework as much as possible before you get into the clinic and you have a very strong clinical path hypothesis. And so you have to bring, it's not just about drug discovery and development, you have to bring the biologists together with the drug developers, and you have to bring the clinicians together to really help the drug developers understand what is the clinical opportunities, what will be the standards of care 5 years from now for particular diseases, et cetera. What will you do in combination? What will be the design of the clinical trial? All of those things are very important before you get into patients. And I'm just going to tell you a little bit, again, because it's a largely academic audience, what we did in MD Anderson Cancer Moonshot. Here, we had reached the point, this was now 13 years ago, where we had a lot of actionable knowledge, but it was a very inefficient system to actually convert that knowledge into patient impact. So what we did is we said we were going to have dedicated money, $3 billion over a 10-year period, dedicated time and infrastructure for this, and we were going to promote team science, break down silos, have professional platforms. This is the most important thing because academics are very good at discovery. They're not necessarily good at translation and doing the things that are necessary to truly validate a target or a drug, right? That really requires almost industry-like execution, right? And then you have to do this in a highly, accountable way with really clear, measurable improvements and so on. And so we started this and we said, well, these were the ones where there was a lot of actionable knowledge. Then we added additional ones. And just in the case of, I heard this just 2 days ago, in the area of ovarian cancer, there were 11 new standards of care as a result of the investment, right? And many practice-changing advances in all of these areas. And then the key thing that we developed, and I think this Academia Sinica has a unique opportunity to do this, is we created these platforms for drug discovery. These are real professional small molecule developers where we invested tens of millions of dollars annually to create small molecules, as well as biologics, so things like CAR-NK. That was a pilot project by Katie Rizvani. We invested a couple of $1 million to get that off the ground. Just the upfronts were $300 million to Takeda, right? And that's an important point because if you make these investments, the return to the institution is pretty significant. MD Anderson, when I got there, its IP revenue, licensing revenue was maybe $10 million a year, $14 million a year. And now, routinely, we're over hundreds of millions. So this is per year. This happens to be 2020, but 5 years in a row, we beat the entire University of California system combined, right? UCLA, UCSF, UCSD, so on and so forth, right? One institution. Why? Because we now created assets that were more mature before we launched a company, right? And what that means is that when it incubates in an academic institution, you take the time to make sure you validate the target, validate the drug, develop the clinical hypothesis for which patients and so on, and then you start the company. If you start the company sooner than that, then it's like premature birth. The baby is always going to struggle, right? And so you really need to do this. And the return means that you can now plow these resources back into your mission and help patients to educate the next generation and so on. I want to just talk a little bit now about the scientific opportunities in the coming decade. And this is more for the young folks. So folks like Minchi and I that have been around the block for some decades now, there's a real palpable turning point in the history of science. This is an incredibly exciting time that I think will go down in history if you look at it over the arc of the last 50 years, since the genetic code was cracked. This is now the realization where for the first time we can actually change who we are, right? This is fundamentally changing the human condition in a very profound way. And so these are just, I think, some of the main diseases that are the opportunities. I mentioned that there were these 10,000 diseases. These, I think, are the biggest diseases, the major unmet need that is going to continue to plague humanity in the coming decades. And I'm going to touch on just, by way of some of my own research, tell you a little about cancer and then about aging. and some recent work that we had done. And really, there are so many ways. When I started my career, there was one hallmark of cancer: cell division. Simplicity. We didn't talk about angiogenesis or apoptosis. That stuff did not exist yet. Now we have 24, right? And but the genome is important, and as we've seen, we have obviously lots of opportunities to attack driver oncogenes like EGF receptor, BRAF, and so on, HER2, et cetera. We have synthetic lethal and collateral lethality. I think that one area that's going to be, on the biologic side, that I would absolutely pay attention to and invest in are ADCs and ARCs. The radioligand conjugates, I think, are going to be extremely area of tremendous growth and opportunity. Obviously, mRNA cancer vaccines are really exciting, and some of the work that's ongoing and from Sloan Kettering and MD Anderson on cancer vaccines and pancreas cancer, really impressive game changers. You've got alternative T-cell checkpoints. So obviously, most of the effort has been in PD-1 and CTLA-4, but there's many other checkpoints, and I'll talk about that. But huge opportunity. The big issue there is you don't know which patient to use which drug in, and Linda will talk more about that. And then we have myeloid checkpoints. If we were going to be doing immunotherapy now, what we know now, we wouldn't actually start in T-cell checkpoints. We would have started in myeloid checkpoints. The myeloid biology is the dominant component in the immune system in the tumor microenvironment. I'm going to talk a little bit about that. And then the microbiome. This is, a continent of biology here that we were missing. It has a major impact on modulating immune responses, toxicities, et cetera, and also the development of cancer itself. So I'm going to tell you about just one story from my lab, and I'm not going to go into the details. But, in pancreas cancer, we, have used CTLA-4 and PD-1, and in combination, 0 response. And there's still, to this day, many clinical trials with PD-1 or Pembro that are, adding additional things, but there's been no response. And so we were curious as to maybe it's possible that it's some of these other T-cell checkpoints. And the other is the myeloid cell. If you look at the tumors have a preponderance of tumor-associated macrophages, of granulocytic MDSCs, myeloid-derived suppressor cells. And so we did a very simple experiment. We actually audited all of the immune regulatory molecules. And for example, what we saw was that CTLA-4, PD-1, TIM-3, LAG-3, OX40 were all present, 4-1BB as well. And then we went ahead and asked, do any of them have responses in a mouse model of pancreas cancer, where PD-1 and CTLA-4 antibodies don't work, right? It's just like the human. And what we found was that when we used LAG-3 and 4-1BB, which is, I don't think, listed here, we saw for the first time some signal of activity. And we used those two together, and we saw even more extension of survival, but all of the animals still died. And what we realized was that there were these granulocytic MDSCs that are playing a very prominent role in the biology of immune suppression in the pancreas cancer. And so what we then did is we used an antagonist antibody to LAG-3, an agonist antibody to 4-1BB, and a CXCR1-2 inhibitor, which hits the granulocytic cells, and got unprecedented responses. These are 3 different orthotopic models, and the orthochronous model will eventually go down, and now in a paper that we have coming out in Cancer Discovery, combining it with KRAS, even in the orthochronous model, where all the animals die within 3 weeks, we have animals, 40% of the animals that are surviving past 8 months. And so now these trials will get started in the coming year. It's just to say that We have a lot of, IO therapies, and we think we know a lot about immunotherapy. This is a simple example, one of the most refractory cancers that it actually is susceptible to immunotherapy. But you have to do it scientifically and systematically so that you can now begin to consider bringing these agents together into phase 1 clinical trials and beyond. So I'm going to just now briefly touch on aging, and this is the ultimate overlord, right? So it's nobody escapes, everybody gets it, everybody dies, right? And it's the basis for all of the different diseases. The hallmarks of aging is something that actually underpins many other diseases. And what's interesting is, just like in cancer, we actually know what the hallmarks of cancer are, and we can target those hallmarks. We can understand those hallmarks. So cell cycle dysregulation, well, we have CDK inhibitors. Signaling, we have, and so on, apoptosis, all of those different, we now have drugs targeting those hallmarks. So the knowledge of the hallmarks has illuminated new therapeutic points of attack. So now, similarly, in aging, we actually know a lot about the underlying components of aging, the molecular aspects of aging. Can we now impact that just like any other disease? And so for the last 30 years, we've been working on telomerase. Nobel Prize was given out for this, Carol Greider, Shostak, and Blackburn. And essentially what they discovered was an enzyme, telomerase reverse transcriptase, and an RNA component that helps to maintain the telomeres. And when we knocked this out 30 years ago, we're able to show that that's really important for health. That is, you get very prematurely aged, and if you don't have telomerase. So maintaining your telomeres, the classical component, is actually quite important. But one of the things that we noticed was that TERT is actually expressed in all somatic cells. And it gets repressed in all cells with the onset of aging. So 40s and 50s a human, 18 months of age for a mouse. And this then leads to a loss of TERT protein levels by just 50%. So it just goes down half. The other thing that we then did is we looked in our hets. So these are young mice, but they're heterozygous for TERT, and they make 50% less TERT. And what we found when we looked at the transcriptome in the brain was that really important genes like brain-derived neurotrophic factor and amyloid precursor protein, two genes that are really important for Alzheimer's, were changed. And that was interesting because, it suggested that TERT can influence gene expression networks. And so we did an experiment years ago, about 15 years ago, where we asked the question, is aging reversible? So what we did is we knocked in the estrogen receptor component into the TERT open reading frame. This TERT ER molecule is inactive. It's like the knockout. So you can generate these aged animals and then give them tamoxifen and renature the protein and turn the protein back on. And what we found was this then led to a reversal of all aging phenotypes. Shrunken brains increased in size, neurogenesis came back, cardiac function came back, metabolism came back, thin bones and calcification increased, muscle strength increased across the organism. Now that was interesting, but what really was curious that every organ system was improved because the classical function of telomerase is to synthesize telomeres, and that happened in these animals. So for proliferative tissues like your GI tract, the skin, the hematopoietic system, that made sense because you have to go through cell division to synthesize the telomere. But we saw post-mitotic cells were also rejuvenated. For example, in the brains, the reason why they're shrunken is because you have less myelination of post-mitotic oligodendrocytes. And now with just 1 month of tamoxifen treatment, they restore the myelination. So this is telling us that TERT must have functions beyond just making telomeres. And what we learned was that TERT actually is a co-regulator of gene expression. It interacts with the mediator complex, and it's able to work together with the transcription factors like beta-catenin to really increase the expression of its target genes. And it's subtle, but it's enough to really change. And these genes are all the genes that are involved in the hallmarks of aging. And in a sense, TERT is a master key that regulates in a very subtle way hundreds of genes that are involved in organ system maintenance. You know, how is it that a stem cell maintains its ability to make 1 trillion blood cells per day when you're still 100 years old? It turns out that TERT is well expressed in those organ systems that maintain cell state. Why? Because they not only synthesize the telomere, which is important, but they also regulate gene expression in a way that maintains cell state. And so we now know that TERT and telomerase and telomeres are linked to all hallmarks of aging. For example, TERT can regulate the expression of sirtuins. Telomere dysfunction can activate inflammation, tissue inflammaging. And just one paper that we published in 2011 actually linked telomere dysfunction to mitochondrial dysfunction with activation of p53, repression of PGC, which are the master regulators of mitochondrial biogenesis, as well as antioxidative defense. And so basically, TERT and telomeres play a critical role across all these components. And so we then asked, well, what would happen if we were to try to control the expression of TERT? And so we did a screen that I won't go into. It's published, but it was a very novel screen where we looked for small molecules, did a 1 million compound screen, that to have this de-repression of TERT. And so with this screen and this unique back transgenic that had a 160 kilobase human TERT locus with a reporter knocked in, we're able to screen all these compounds. And we're able to show that compound, one of which is shown here, we have quite a few others, is able to subtly restore TERT expression. And it's able, in vivo, this compound is able to increase expression in the hippocampus, the cortex, skeletal muscle, heart, and so on. And then we did a series of experiments. So these are very old mice, essentially the equivalent of a 75-year-old. And we treated them for 6 months, essentially the equivalent of a 95-year-old, with TAC, so just 3 times a week. And we saw a couple of really interesting things. We saw new neurogenesis in the hippocampus, which is very important for working memory. We also saw a dramatic decrease in senescent cells. So this is just SA beta gal. So all tissues, we eliminated senescent cells. We also reduced dramatically inflammatory cytokines in tissues and in circulating cytokines, so inflammaging. We looked in the brain, and we saw that there was a tremendous decrease in neuroinflammation, which occurs in the aged brain, occurs in Alzheimer's as well, chemotherapy, chemobrain, and so on. And we see a dramatic decrease in key cytokines, particularly TNF-alpha, which is a master regulator of inflammatory cytokines, and so on. And if you look in the hippocampus of animals that were treated versus the controls, we see things, like telomere maintenance, which makes sense. We're activating TERT. But, neurotransmitted metabolic process, dopaminergic neurogenesis, stem cell, and the only thing that went down was inflammation. And so this drug is able to reprogram the cell state of diverse cell types in a way that is very beneficial, essentially rejuvenates the animal. And so that is consistent with the fact that in 3 different behavioral tests, cognition was dramatically improved. Again, I won't go through this because it's published, but the escape hold test, the novel recognition, Y-maze, they all scored very highly. These are, again, these very ancient animals, the 28-month-olds. And also we saw that if you look in skeletal muscle, so sarcopenia is a huge problem, decreasing muscle mass in aged individuals. And we saw things like myogenin, MyoD, and so on increase, these master regulators of muscle function. And we see that grip strength was increased, and also balance, speed, and coordination was improved as well. And so just to close, this is an example where basic science, not focused on anything that is necessarily translational, but just really trying to understand what are the hallmarks of aging, what are the mechanisms behind those aging, and the fact that actually TERT and telomeres play a really integral role in all hallmarks of aging, put us in a position to be able to then ask, can we reprogram cells in a way that could really change the somatic state, right? And when you think about it, we were trying to figure out this idea of like how does a 100-year-old hematopoietic stem cell still make all of those blood cells, or your GI tract is still turning over every 7 to 10 days and so on. How does that happen over such a sustained period of time? Well, a lot of folks have really been interested in, well, maybe it's some epigenetic regulator. But the epigenetic regulators across species, across different stem cells, across different tissues are all different. They all utilize different things, but epigenetics is important. And so one way we're thinking about this is that these diverse epigenetic regulators actually control the expression of TERT. And so if you can reawaken TERT itself, you can then really make an impact on genes that are really essentially trying to maintain a more youthful somatic state and potentially impact the aging process very directly. So I just want to close with a couple of points to bring everything together. First and foremost, what you're embarking on here to create a biotech industry, a biotech ecosystem, is not easy, right? This is a very difficult, it's going to take the best in all of us to be able to make an impact. And it really requires not just money, it requires collaboration. Collaborations that don't normally coexist in a coordinated way. But if the VCs, government, academic, basic science, academic institutions, and very importantly, clinical programs come together in a coordinated way, you can really accelerate, leapfrog to a very vibrant program that can really make a tremendous amount of value for patients and for investors. And a very important aspect from a national perspective, and I hope that there are maybe some government leaders here. There is, again, a very direct correlation between vitality and security for those nations that invest in research, basic research. Think mRNA, right? GLP-1. Those are all fundamental science. Jim Allison, when he discovered CTLA-4, he was just interested in how T cells work. He was not thinking about curing cancer, right? And so this is very important, that the government really needs to put the wind in the sails of its institution and its talent. I would say to the students, I wish I was starting my career now. This is an incredible time. I'm sure, well, I started when the molecular biology revolution was starting. We can clone and sequence genes for the first time. It was so exciting, and it changed things. I think that there is now a similar tectonic change. Maybe I shouldn't say tectonic in the ring of fire here, but a tectonic change, with artificial intelligence and so on, the ability to design things. One of my biotechnology companies developed a drug in 9 months, a clinical candidate in 9 months with $1.5 million. Now, industry average is about 14 million, and takes about 20 months, 24 months. Why? Because we used, structure-based design and AI, right? It's absolutely going to impact everything. And so when we look back over the last century, people 10,000 years from now are going to be looking back as a major turning point in the history of science and who we are as a species, right? So obviously, the atom, discovery, of the atom and cracking the atom, the chip, obviously, and the structure of DNA that really changed this. But now, in this century, the atom is given way to quantum physics, the chip is given way to artificial intelligence, and the gene has given way to synthetic biology, right? The ability to edit the genome at will, right? Think sickle cell and what's happening there. These are all very, very exciting times. And it's really critical that we get our act together and we do this, because we are careening towards a very significant issue. Because, when around the time that I was born, in Asia, the average life expectancy was low to mid-40s. And today, it's approaching 80, right? And I mentioned after age 60, you have this geometric increase in these very costly diseases that are going to just crush governments. And so we need to be able to invest to maintain our health span. Now, whether we can change lifespan is a different question. But health span is something that I think we can, with the knowledge that we're accumulating, not just in specific diseases, but of the aging process itself, where if we can increase that health span, then we have an opportunity to really make our societies far more vibrant, maintain the vitality of our aging, wise individuals like Minchie, and to really, I'm getting back at the introduction, and really change this. So this is, our solemn responsibility, right? It's not just about, no one's going to remember who we are 100 years from now, right? They might remember, Jim Watson and folks like that, right? But really Louis Pasteur, right? So our responsibility is to come together as a community to make an impact on pain and suffering. And if we don't do it, and we don't do it together, it's not going to happen, right? So you really have to work together, and it's not just working together collaboratively within the academic institution. Academia, hospitals, government, VCs have to come together, and I think what this country can do that, because it has a history of doing exactly that. It's a model for the world. So I just want to thank, I mentioned some of the work that, I just want to point out one individual here, which is Hong Suk Shim, who did the TAC work, the telomerase activator compound work, and Young Hong Liu, who did some of the pancreas cancer work that was started by Pat Gulhati when he was in the lab. And of course, our collaborators, we work with Pete Schultz at the Scripps to do the screen, and we've been supported by wonderful organizations that have put the wind in our sails, and my trainees to be able to make an impact in a small way. Thank you very much.
I assume we might have a couple minutes. Okay, but I want to make one point that at the end, collaboration. So we all have to remember we need a collaboration. And then, okay, you have a question now, so I'll let you go first. Yeah. I want to say everybody the last message. What, 1.5 million, 9 months, go to clinical trial. Okay.
And I think it's very important for this audience. And first of all, I'd like to thank you for this very inspirational speech. Personally, I learned a lot. And one of the key points in your about this ecosystem is you deliver a message very strong, and I'd like you to elaborate a little bit more, is that don't start a company too early.
Yes.
Can you?
Well, it's kind of like having a birth that's in the second trimester. The baby's always going to struggle for the rest of its life. See, because if you can, the private sector, you're on the clock, and if you don't have the infrastructure that you have or the patience and time. You can fail in the clinic. You can fail in academia. In fact, most of the things we do fail, right? You could take the time to really mature the assets. And that's not only reduces the rate of failure later on and increases the probability of success. That's very, very clear. So for example, if I think Dr. Chin will mention this, but if you have biomarkers associated, you have a 6-fold increase of success, right? So that only gets really developed when that asset, that drug and so on, when you still are able to collaborate with your academic colleagues and your clinicians and so on. As soon as it becomes a company, an iron curtain comes down, and it's very difficult for that company to really seamlessly interact with collaborators as needed. And if you bring this to a mature asset, more mature birth point, the institution is created more value, and when it gets licensed into the company, the return on investment to the institution is going to be significant. Because MD Anderson invested in the platform of drug discovery and development, but also did the biology together with the clinical, the assets that we created were much more mature, and when the company was started, the companies were more likely to be successful, and the licensing deals that Anderson was able to secure was much better. So it helps everybody, and it reduces the most important thing, which is failures in clinical trials, which are very bad for the patients. Right? You're subjecting patients to failed clinical trials.
Yeah. So you want to nurture the company, the technology nearer to IND before you invite private sector to found a company.
I would say
so there's an iron curtain.
The hallmark,
start there.
I didn't get into a lot of this, but, and I have a website here that I have, that's over here, that I've written things about starting a company and so on. So you could just look at that, and it's just stuff, because I talk about this all the time. But it's basically a really good company is a company that has a development candidate that's extremely well credentialed with a clear hypothesis, maybe a couple of other assets to mitigate risk in case the lead program fails, and maybe has a platform. That's like the ideal golden, child, right?
Really wonderful talk. And it's just regarding scientific question regarding the TERP, essentially it's a master switch for the aging, right? So I just wondering whether you can make some comment about Michael Snyder just published last month regarding this proteomic study over 100 people, and he found that the age mid-40 and the early 60 have this a 2 point for the aging alter the metabolism process. So I was just wondering whether TERP, whether it's really age dependent, age onset, at mid-40 or early 60.
Yeah, well, it depends on the lifestyle. So if you have a very poor lifestyle, you see aging hallmarks like senescent cells, inflammation, and so on, starting more closer to 40. And if you are, exercise and take care of yourself and so on, it tends to happen in the 50s. So there's a kind of a biologic age gap as opposed to a chronologic, your age versus your biological age and so on. And that there's a wide range. In mouse, it tends to occur right around 18 months of age because they're all more or less the same. A little bit different in different strains, but that's good question.
Okay, so while we're waiting for the question, can I ask a quick question? Tech, appearing by whatever criteria to help the aging, prevent it from aging. So I was wondering that you already, is this tech compound transcriptional upregulate TERT or enhance TERT activity?
So we're still, working on the target, and we're not talking about that yet until we know for sure. But we think we are understanding the complex that it interacts with that ultimately leads to activation of AP1, which serves as a pioneering factor that de-represses the TERT locus, right? So it's activation of AP1, and so that AP1 serves as a pioneering factor, And that's how
Okay,
you
so-
de-repress TERT, and that's enough to increase TERT back to youthful levels, and then the cascade of events after that.
Okay,
Can I have
thank
one
you.
more question?
Okay, yes.
Yeah, it's a very inspirational speech. I'm curious, you mentioned that the AI accelerates the whole process. What is the most optimal mode for your interaction of AI and your researchers? And how much investment that you have put into that kind of building that AI expertise?
You can't, I think obviously there are companies, and also it's important to appreciate there's a lot of stuff you can outsource. You don't have to build everything in-house. So Schrodinger, for example, has a terrific platform that I would encourage the students to learn about and so on. It's not just in drug discovery and development, but even the way that Dr. Chin developed the biomarkers for patient selection, that was also AI-driven and so on. But it's really, AI is affecting everything. I would say that AI is probably equivalent to electricity, right? The advent of electricity. It's going to have a huge impact on everything that we do.
So as early as possible to introduce AI in the whole research process.
Yeah.
That benefit.
Yes.
Even broadly.
All aspects. All aspects. Transformative.
Okay, thank you. Oh, okay, Alice. Okay, there are a lot of. After Alice and you. Okay, so go ahead.
Thank you. I have a quick question about the phase 3 clinical trial. As you know, failure in phase 3 is very, very costly. Now, in recent years, especially for orphan disease or rare disease, the ODAC or FDA has allowed the use of ECT, external control trial, instead of randomized trial, to facilitate the drug approval, or even real-world data. I'd like to know what you think of that.
I think, for early stage, for rare diseases, I don't see any alternative, right? These drugs would never get developed, right? And the return on investment is also a challenge, right? So you can at least get it into the market and see. I think that, my view is the FDA, for example, with the Alzheimer's, that's not obviously a rare thing, but what they did to approve the, amyloid antibodies was courageous, because that kept momentum in the field. Even though it was not an optimal drug with side effects, it allowed, it continued to, it continued to keep the momentum for the field and so on. So we have to look for ways to incentivize, especially pediatrics, for example, the rarest of the rare, think sarcoma. I think it's very, very important that we do things. Here's actually a phenomenal opportunity for institutions around the world to work, right? If you had like leiomyosarcoma, but if you had, it was a worldwide clinical trial, you could then do something meaningful, right? But, hundreds of cases per year in the United States, you're not going to go anywhere.
Okay, last question.
Okay, so thank you for the very nice talk. So it's a scientific question. So I think one of the key idea that I learned from your talk is that we are not looking for to extend the lifespan of human, but we are looking for the health span of human. So in this term, so you come out the rejuvenation. I think that is a big dream for whole human beings. So in your study, have you ever looked at check the mitochondrial activity after treatment?
Yeah, so that's very important, and I'm embarrassed to say that we haven't in post-mitotic tissues. In mitotic tissues, we reported in 2011 that it plays an extremely integral role in mitochondrial biology. Regulates virtually all genes that make mitochondria and make mitochondria work, right? So there's a very intimate link. But we have not looked at the ability of TERT to regulate genes that are involved in mitochondrial function and so on, or other things like oxidative defense. So very important, that's work that's ongoing. And so I do want to make one point, which is, the field of aging has been kind of marred by folks who make a lot of hyperbole. And, as a scientist, I have to say that very provocative studies in mouse. We don't know if it's going to translate into the human situation. We have to do this, we have to do the experiments, do the trials, bring the right drug forward, the right schedule and dosing, and so on and so forth. So it's something that's going to take time for us to really sort through. The FDA has never done an aging trial, right? Our idea is like GLP-1 is to start with diabetes. Oh, they lose weight, you do weight. Oh, the cardiovascular risk. Oh, it even helps addiction. Right. So what you can do is you can start with specific diseases, sarcopenia, for example, and then you start to build more and more knowledge of that. So if it does translate, it's going to be helpful. Now, lifespan is linked to metabolic rate, which I think is what you're alluding to. And there, in order to live longer, if you look at C. elegans, you know, the work of Cynthia Kenyon and so on, to live longer, you just have, you turn the oven down, right? You cook slower, right? Metabolism, you slow metabolism, right? And that is the basis for life extension. So metabolic rate is hardwired into species, and that is kind of the main parameter that dictates lifespan, right? So we haven't actually done any lifespan studies with TAC. I think, you know, mice would live longer because they're not going to die of some of these other issues.
Volunteer test. Yeah, okay.
You and I both.
Thank you very much for a very inspiring talk. So I'm wondering, for institutes like MD Anderson, you have a clinical center along with strong research scientists. So it will be relatively easier to do clinical trials. What if some universities, they don't have medical school or medical centers in tight collaboration, or no medical school on campus? What would be your suggestion? Like how do we like Academia Sinica, there is no hospital. So how do we attract credential hospitals with clinical research teams to collaborate or support us without sharing IP way in the beginning? Sorry.
Yeah, no, I understand. I think, you know, first of all, I do believe, and again, you're looking at literally thousands of examples at this point. If you do not bring your clinical insight early on, you will have a very high rate of failure, and you'll have 100% ownership of nothing. Okay? So it's important to appreciate that the more you work together and invest, your skills and knowledge, it's not just making the molecule. Actually, I would argue that it's not just the composition of matter, but it's the label. What is the indication? And I can tell you there's many, many drugs, great drugs, that the wrong fight, look at all the immune checkpoint inhibitors. They're all failing because the 85% that don't respond to PD-1, like who has LAG3, who has TIM3, who has whatever, right? Which patient do you list in the clinical trial? If you knew that, your LAG3 antibody would be extremely important, right? That would be key. So you need to have all these elements working together. If you do not, then you're going to be subject, you could get lucky, but you're going to be subject to a very high rate of failure.
Yeah,
okay,
appreciate.
Apparently, but the time is running out. We have a more than 15-minute discussion. So the last message, again, is collaboration. Hey, I see that you're a hospital. You have a hospital, right? There are many people in hospital. Taiwan had to work in together as a team. We cannot work separately. So I think that I would suggest that we give Dr. DiPino another big hand. Thanks for his sharing.
Thank you, Dr. DiPino. 同时也谢谢我们洪校长为我们担任引言人，而且也为大家呢追加了这么多的问题。Thank you again. 那接下来呢，我们就直接要进入午餐时间。Now is the lunch time。我们今天的人数众多，所以午餐时间因为场地的关系，我们会分流用餐。所以麻烦帮我呢注意一下，今天识别上面的袋子颜色有所不同。如果是粉红色袋子的，各位来宾，请帮我跟随工作人员的指引，帮我来到呢 A330转译学研办公室用餐。所以如果是粉红色挂绳的来宾，可以跟随我们的工作人员，在舞台区的右侧前往 A330办公室用餐。那如果您是黄色吊绳的贵宾，黄色吊绳的贵宾，请帮我来到 A215的贵宾休息室用餐。好，所以再麻烦大家帮我注意，粉红色吊绳来自 A330，黄色吊绳来自 A215。而我们在3楼的大厅也有准备大会的餐点，所以所有的来宾都可以来到3楼的大厅来进行用餐。那同时提醒，我们在1楼也有为大家准备茶点，同时也展出了本次的参展团队，共是60多组的团队，都在1楼有海报区的展出，以及此次活动的赞助商摊位，都欢迎大家可以多多的莅临参观。那我们下午时间会在1点半准时开始，再请各位来宾提前5分钟回到会场。我们1点25分再次回到会议场。我们下午的议程会在1点半准时开始。祝大家用餐愉快。 们现场所有的长官还有来宾，欢迎大家回到下午场。Nbrp Pitch Day 的活动现场，我们的议程即将在1点半准时开始，目前还有2分钟的时间，所以邀请还在场外的贵宾，请帮我把握时间入席。已经入场的宾客，麻烦也可以先找到位置坐下来。那还是要邀请大家如果可以稍微集中入座，可以往前集中入座，让我们现场的座位看起来更热闹一点点。那我们的下午议程第1场的专题讲座呢，马上会在1点半准时开始，再请大家把握时间入席，谢谢大家的配合。各位现场的来宾朋友，大家午安，大家好，欢迎回到我们下午的议程现场。下午的第1场专题演讲，我们将在2分钟之后正式开始，邀请各位贵宾把握最后的时间入席，谢谢大家的配合。 Hello， 现场的各位贵宾以及各位来宾朋友，大家午安，大家好，欢迎大家回到2024 Nbrp Pitch Day 的下午议程现场，我是今天主持人 Penny。感谢大家准时的回到现场，我们接下来即将进行今天的第一阶段演讲，而在第一阶段的演讲结束之后呢，紧接着我们就会进行今天新药技术发表竞赛的决选，所以欢迎大家呢和我们共襄盛举。那今天在下午的第一场演讲呢，我们特别邀请到了是来自中国医药大学的校长洪明奇院士哦，他们要为我们带来呢关于精准医疗的解说 ，Utilizing Semer Markers and Diagnostic Technology for Optimal Treatment。那在邀请洪院士为我们演讲之前，我们要有请中央研究院的陈玲金院士为我们担任本场演讲的引言人，掌声欢迎陈院士。
Thank you. It's my distinct pleasure to introduce to you Dr. Ming-Chi Hong. I have known him for a long time. Before I go into his scientific contribution, I want to say that he's a remarkable singer, well known for his rendition of Elvis Presley's timely classics, timeless. Okay. Ming-Chi Hong, as we know, is academician of Academia Sinica and also the president of China Medical University since 2019. He is a graduate from National Taiwan University in chemistry and followed by a PhD in molecular biology at the Brandeis University. I know he spent at least 20 years at the MD Anderson Cancer Center as a professor and the director of molecular cell biology and all the way to the vice president of MD Anderson before he came back to Taiwan 5 years ago. I don't think I will take too much time about his amazing scientific contribution. Suffice it to say that he's not only one of the pioneers for the tyrosine kinase receptor tyrosine kinases, signal transduction, cancer biology, and also an entrepreneur in many of his inventions and with successful industry transfer. Dr. Hong, please.
掌声欢迎洪院士。
Well, thank you, Alex. Great pleasure that come here to see a lot of our friends. Especially, I'm very nervous that I was presenting in front of my boss, and also even more serious in front of my boss's boss. I think at home, I assume Linda is the boss, right? Okay. Anyway, so first slide. I think to save time, I'll just go ahead. Anybody doesn't know him? By the way, Jensen Huang came to Taiwan very often, and he liked the night market. You guys, did you go? Did you take them to night market? You take them to night market, you might see Jensen Huang hanging around there. Yeah. Okay. So Jensen Huang said that the computer time is past. So now, it should be life science. It's the future. I don't think he, come on, his company, the AI company, he need to make money. I think he just tried to make him look better, that he said life science is important. However, to say that, life science doesn't matter, well, we all have a life, well, Ron just mentioned about the aging. So life science is always important, regardless that it's like molecular cloning or regardless it's chip technology or AI technology. Life science is also important, always very important. And this morning, we just heard about, the first emperor of Qin dynasty, Qin Shi Huang, he was the one who built Great Wall. Everybody know that. But his dream he has one very important dream he's never come true. After 2,000 years later, because he was looking for 长生不老药, he was looking for some magic pill that he can live forever, but he never really success. After 2,000 years later, and we just heard from Dr. DiPino's talk, he might get it. Hey, those me people at the lunchtime, you have a lot of money, you want an investment? Okay, better talk to him wrong. Tech, that one, he has a lot of very interesting data. Okay, so anyway, so Ron, you have a now new title now. The first emperor of Qin Shi Huang. Okay. Okay, so I want to share, we'll talk about cancer today, that I want to share with you some of the global and US and Taiwan's data. That, by the way, US data is from USA. But global is, depending on how you define the global. This is from WHO. WHO global is incomplete, but later on I'll share with that. So if you look at the incident rate, US, of course, did reduced, and but mortality rate in every 100,000 people, male, female, actually all looks pretty good. I think we all should all feel very proud, and especially people working in some, such as MD Anderson Cancer Center. Linda, Ron, and myself too, we feel good because the cancer mortality rate is significantly reduced. But, well, when you say global, Taiwan is not in, okay? WHO does not include Taiwan. So this data had to come from Taiwan's government data, okay? So if I just look at cap out the same time, incident rate still increased, but the important point is the death rate, the mortality rate here. And by comparison with the US and world, I just kind of put it on another slide, okay. Here's Taiwan, male, total, female, out of 100,000 people, the mortality, how many people die? In the US, much less. Hey, you live in the US, you have less chance to die. Male, female, and even world. And this world is not like the real whole world, okay? That doesn't count China, India, Africa, whatever. And here is the data included from WHO. So I don't know why they do this way, but anyway, that's the way it is. So for people living in Taiwan, well, virtually everybody is living in Taiwan except Linda and Ron, right? So you look at this data, and this data, and this data. It's scary, but luckily, our new government have decided to put what, 5 billion NT, right? Every year. I hope that the money really come, and then that we could. And earlier we discussed that we had to work together to improve a lot of area, but of course cancer, because our death rate, our mortality rate actually pretty poor. Okay. Now, I want to turn that today we talk about Pitch Day. We talk about a lot of biotechnology company. So 20 plus years ago, 2000, this is the top 10 drug in the world. And this is $1 billion, $6 billion and all these drugs. And you know, you look at it, this drug, except for this one, which is protein, right? We all know it's a protein drug. The rest, all small molecule in 2000. But today, 20 years later, that this is the 2022 and the 2021 year, put different color, and this is the US dollar billion, that, well, the drug price is increased. But if you look at what kind of drugs it is, small molecule still have 4 of them. But the rest, there are another 4 antibody, and of course, because of COVID-19, this mRNA vaccine came out. I think what this data tells us is not because the small drug it doesn't work. No, it's still working, but the new technology coming out. And this new technology coming out, they're resolving 2 things. One is price is increase. Well, it's not good. But the other one is there are a lot of new drugs coming out in the top 10 seller now. So of course, this is because vaccine and all this antibody and 4 small molecule. Now, I want to share that using antibody as an example. 20 years ago, there's no top 10 drug seller from antibody. But today, 4 out of 10 drug antibody seller. Antibody, we all remember 3 of them developed hybridoma technology. 1975, the big event when we were young. And 9 years later, academic usually take about 10 years. Being recognized, they received Nobel Prize in 1984, 9 years later. And at that time, many people, there are a lot of hype because anybody can be a drug. But look at this market value from 1975 all the way to 1998, this 20-year period. There are a lot of money, especially for those people who are senior at that center code, a lot of money there. Okay, it doesn't work. Because every time when a technology, the concept developed, by the time when it's become a real drug, there are certain type of technology we need to improve, and that's what happened in the 20 years. But after that, the monoclonal antibodies started being approved by FDA, and now the market value continue to increase, including the earlier we mentioned Jim Allison and Hongzhou and the other drug. And then based on estimation, next year is $300 billion. This including what? Monoclonal antibody, bispecific antibody, trispecific antibody, and earlier Dr. DiPino mentioned about the antibody drug conjugate. There are a lot of new way to develop drug. Antibody drug conjugate, if I remember correctly, in the last 3-4 years, there are 11 drugs using antibody drug conjugate was approved by US FDA. I believe that's for technology-wise, it's been approved by FDA. This technology is probably the highest density to be approved. And 11 drugs in 3 years, US FDA approved. And I think there are a lot of drug cooking right now. So this is telling us the market value is so big. And well, Han Chong, you were there, okay. So how much this Nbrp is going to contribute among this percentage? Okay. It's important, okay? So this is KPI. And when I came back to Taiwan, by the way, it was nice that in US, in NBN, you are so nice that you don't tell us KPI. But when I come back to Taiwan, everything is KPI. Right? I mean, I didn't even know what is KPI mean, but when I came back to Taiwan, I know what is KPI. Everything is KPI. Okay? Okay, so then that's antibody. How about let's take a gene therapy, because the mRNA therapy that the COVID-19 has been. So from the very, very beginning, the double helix was discovered all the way to more recently that, well, remember gene therapy, that's in 1999, that this unfortunately, that Jim Wilson's clinical trial in UPenn, that Jesse Jessenjoe, the 18-year-old kid who was healthy, but during clinical trial passed away. That particular trial actually holding the gene therapy for almost 1 decade. But now, luckily, that 2012, CRISPR-Cas9 developed, and then later on, they received a Nobel Prize in 2020, and then mRNA therapy, we all know that COVID-19, that now last year they also received Nobel Prize. And this year, we know the US Alaska Award and also Taiwan's Town Prize, those awardees, they are hanging around in Taiwan right now. They are also, like us, they are waiting for typhoon coming now. Tomorrow they're supposed to go to Taichung. I hope they can go, but anyway, they are waiting for typhoon to come. And those are people who develop GLP-1. What I want to share with everyone is all this new technology, antibody development, which is market value is $300 billion next year, and mRNA therapy, gene therapy, all this technology all become more and more mature. I still remember a while ago we talked about mRNA and nobody wanted to do mRNA because RNA is very unstable. But now mRNA therapy turned out to have a lot of advantage to develop technology. And I always like to show this slide. That is, Tom Price from Taiwan recognized 3 of them. But 1 year later, Nobel Prize recognized only 2 of them, and doesn't include that. It depends on the people's opinion, okay? Because he developed the nanoparticle, and both of them, well, we all know she's interested into mRNA, but he's an immunologist. Well, he was from Brandeis, so we overlap at the same time, although at that time we don't know each other, but now we know each other, okay, because he got a Tom Prize and Nobel Prize, okay? So these people, the reason I want to show this slide, it's the point that we earlier been mentioned every time in this talk, in this presentation, that is 3 of them collaborate. Not 3 of them collaborate at the same time, but at a different stage that this person collaborate with the other person, another person collaborate with another person. Because of their collaboration, that at the time when SARS-CoV-2 hit people, COVID-19 become very serious and pandemic disease, because they are already using the mRNA vaccine development. So it's not because the COVID-19 comes so that suddenly 1 year a vaccine is developed. No, the technology is already there. They are already developed the technology. They are doing the clinical trial. But once the SARS-CoV-2 come in, once the sequence was published, they can change the project quickly. And therefore, today, we are actually pretty good, be able to get together in the same room, and most of us did not even wear masks. If they don't collaborate, today, if we get together, probably everybody wear a mask. Or we probably cannot get together, we probably online. Probably online. So I want to use this slide to emphasize how important collaboration is, because for those people, if they don't collaborate, forget them. They will not receive Tom Prize, they will not see Nobel Prize. Okay? Now, 3 of them, because they developed GLP-1 for treated diabetes or obesity, that they received Tom Prize this year, and 2 of them are in Taiwan right now. And a couple of weeks ago in US, Lasker Award gave to 3 of them. And these 2, the same person. But then, again, it's depend on how committee review things, because all these awards maximum 3 people. So, and they are the people who have significant contribution probably have 5 or 6 or even higher. So therefore, people had to narrow it down. Okay, so I should say, if these 2 are not collaborate, Novartis will not exist, because when they develop GLP-1 antibody, he's a physician. He has been working on that, but he's a peptide chemist trained by Mary Field that to make that peptide really working. So today we don't have to talk about scientific detail, but without their collaboration, the drug is not there. And he's from Europe, and he's independently doing something similar, although his paper was published 1 year after their paper. Okay, so again, I just want to emphasize collaboration is very important, especially Taiwan. How big is Taiwan? Taiwan in the map is very tiny. What the population? 25. How many? Compared to China, compared to US, forget it. We don't collaborate, there's no competition power. I think I just want to emphasize this point. And now I want to come to a little bit science. Those are something that ongoing. That all of this project was initiated when I was at MD Anderson, and 5 years ago I came back to Taiwan, and so I brought all of them back to Taiwan, and currently it is ongoing in Taiwan. Okay, the beautiful lady, who did not watch her movie? I like to watch movie. I watch a lot of movies. Do you watch a movie? No, you don't watch a movie? Do you watch a movie? Anybody did not watch her movie? Anyway, we all know her, okay? But in 2012, 13, she made a big decision. She removed her breast. Movie star. And later on, she removed her ovary. Because she was diagnosed with her family, she has genome mutation of BRCA1 gene. And today, we don't want to talk about a lot of science, but just to quickly, briefly say that in our human body, there are two different types of DNA repair system. One is hormonal recombination, which absolutely requires BRCA. So once this one was mutated, then your allele, one allele from daddy, the other allele from mommy, right? One allele, you still have a mutated, but you still have allele wild type. However, the wild type allele was mutated, then that particular cell are very easy to be mutated to develop breast cancer and ovarian cancer. And another pathway to do the DNA repair is related to pyrylation of PARP1 enzyme. So once these two pathways can respond for DNA repair. However, if this family BRCA was mutated and then developed breast cancer because BRCA was mutated, this particular pathway disappear in that particular cancer type. However, in the normal cells, still have one normal allele. Therefore, at this moment, you can use so-called synthetic lethality, use an inhibitor to block this one. So you block selectively the cancer cell, but not normal cell, because normal cells still have one allele, okay? So this was done now, 2013, but currently there are at least 4 drugs, maybe more now, being approved by FDA. But not all of them 100% recover. They're usually 50, 60% response rate. So there are some resistance. So here I just want to quickly share with you that we try to identify the mechanism of the resistance and develop marker to identify a specific population of patients who are resistant. Therefore, you can use a combination therapy to overcome resistance. And this gene was originally identified by Mary Clary King, and she came in a few months ago, came to Taiwan. And Ron and Linda, remember Neil Towino? Now I've been very nice to him because he's the cancer center director in Hawaii. In Hawaii, so I've been nice to him. So next year summer, I'll be hanging around in Hawaii. So he was from MD Anderson, but now currently he's the University of Houston Cancer Center director. So they were visiting in the conference, so we took a picture. And Mary Clary King is the one who developed to identify the BRCA gene. So here I want to share with you our approach. And I have been working on receptor tyrosine kinase, not because the receptor tyrosine kinase is important, it's because it was earlier being discovered and there are a lot of FDA-approved drugs attacking receptor tyrosine kinase. So if you identify a mechanism involved the receptor tyrosine kinase, likely they're already FDA-approved drug. So you can quickly to take that drug to help patients. So the way we do it is we took the sensitive cell. This is a very sensitive to PARP inhibitor drug, but they are BRCA mutation. And use the antibody array. So these two are resistant cells which we develop. And by this comparison, you can quickly, between this sensitive cell and resistant cell, all these are control spot. But you can see this spot, light up in resistant cell, but not in sensitive cell. This experiment tells us these type of receptor tyrosine kinase are likely, not necessarily, likely involve the resistance. And after that, we can test them one by one. So there's a series study from our own group and from our collaborator together that we have now identified all these tyrosine kinase has ability to cause resistance at the different cell line, and each one of them we have the cell line. Okay, so some of them published, some of them is still ongoing, but here I just quickly want to share with you that our first one, which we identify, that is every resistant mechanism, we always identify a specific marker, especially a novel phosphorylation, for example, so that we can use this marker to identify patient. So in this article, and he's still in UT, he's not in MD Anderson now, but he's in UT Health Science Center as a faculty member. So what we discovered is receptor TNFC, triple negative breast cancer, CMET was overexpressed 30%, and c-Met can phosphorylate PARP at this particular tyrosine site, this is a novel site, and after that become resistant. And the reason it become resistant because the mechanism is, then once this PARP enzyme phosphorylate in this site, they reduce ability to bind to binding this inhibitor. So therefore, the enzyme activity was reduced. Okay, so with that, we then use the combination therapy for PARP inhibitor and c-Met inhibitor, which already have FDA approved trial. That's why we're interested in tyrosine kinase, because a lot of kinase inhibitor already available. And so it showed very nice result. And there's a clinical trial actually was done by Tim Yap in MD Anderson. And so here we have one marker. Very similarly, another article, then she's still in MD Anderson. We identified another marker, CDK9, which can be tyrosine phosphorylated at this particular site by LK. And LK is a receptor tyrosine kinase. And once the LK, which a lot of cancer type, this gene was either mutated or overexpressed, phosphorylate CDK9, then CDK9 can move from cytoplasm and go to nucleus. And this is already known by binding to the second T to turn on a lot of transcription, activate POR2. That's already known. What's not known is what they turn on are all hormonal recombination downstream gene. So even if you have a RCA mutation, they don't care. Because once you have this pathway activated, hormonal recombination still occur. Therefore, it causes resistance. Now, then you can, in principle, that you can use the LK inhibitor plus PARP inhibitor to the combination, and that's the animal data, survival, that without treatment, one drug treatment, the other drug treatment, but when you combine together, looks pretty good. So that's the other one. And again, as I mentioned, every one of them, we have a specific phosphorylation marker to identify. The third one, all these are all very similar, but one by one, this was still under a revision, that we identify PARP tyrosine 158, another novel phosphorylation site. After this site was phosphorylated by FGFR receptor, you know what happened? PARP will be able to bind into DNA, then after phosphorylation, they will leave to allow the DNA repair to continue, and therefore cause resistance. Okay? So then with this phosphorylation site, and with FGFR receptor inhibitor also has been FGFR, this one has not been approved yet by the under clinical trial. Then this type of patient, if you have this marker, you know they are resistant, you can use a combination therapy between PARP inhibitor and FGFR receptor. So I think one by one, what I want to share with you is that by knowing in the lab, by knowing the mechanism, identifying the marker, we should be able to identify right patient population to be treated the right combination, and those is the right combination. Many of them already have FDA approved drug. And this is another one just came out this year, Gasman C. Well, maybe I don't have to describe the another detail again, because this is also published this year, that essentially what we say is Gasman C is a molecule is known to be able to form a channel in the cell membrane, and Gasman C can induce, when they are activated, it can induce paraapoptosis. And what we found is then this Gasman C in the immunocompetent mice go to this mechanism, actually enhance fever-for-war mechanism, enhance the PARP inhibitor to inhibit. Therefore, Gasman C high tumor may also be able to sensitive to PARP inhibitor and so on. So with all these, and whatever we do, the bottom line is patient. And this patient with a very unusual case. This is a head and neck patient. It's a huge tumor here. And when I talked to, 2 years ago, when I talked to my collaborator in China Medical University, he is his patient. And he told me he's going to die because his condition is so poor that he could not even walk up from the bed. And he said he's in early 22 January, he told me he's going to die. And we talk about collaboration on the other project. Then I asked him, well, what kind of drug he was treated. He was using it because he was a BRCA mutation. He was treated with one of the PARP inhibitor. It was resistant. So I told him, well, since we have a list of the kinase, those we know could cause resistance, some of them are not published yet. But then he went to dig out the data to find out one of the kinase turned out to be was mutated, activated in this patient, and therefore it doesn't work. So I told Dr. Xie, I said, well, do you want to try the kinase inhibitor? And that kinase inhibitor, luckily happened to be available in Taiwan. And sometimes some of the kinase inhibitor, some of the drug available in US doesn't mean it's available in Taiwan. Okay, so he was lucky. So then, since he's going to die anyway, the condition is very, very poor. So they were tested. The first round from January to March, we thought the tumors drop. We thought he's going to alive because he can leave the bed, can walk, can exercise. So we were so happy that he may be one case, although it's one case, it's no statistical significance. It's just one case, but anyway, one human data. But unfortunately, he expired in June because of the complication from other things. It's not because of the tumor by itself. But these data are actually very encouraging, although only one patient data, but at least we have one patient data. So what we would like to do is following, is an umbrella clinical trial. So I just mentioned all of this, from the public, I quickly mentioned, we all have a marker. And we all know this marker causes resistance to PARP inhibitor. So if those patients who are BRCA mutation come to the cancer center or come to the hospital, then if we can use a mini antibody array, which, oh, by the way, by all of these specific novel phosphorylated antibody, we will induce monoclonal antibody by ourselves in Taichung. And so we should be able to use the mini antibody array and then take the tumor, small amount of tumor tissue before they will treat the PARP inhibitor to see whether they might be resistant. And if they are resistant, which particular molecule cause they are resistant. And if we know that, not all of them, but significant portion of them, we may be able to use directly use the combination therapy. So that was what's the goal. So this currently is ongoing. And the trial will start in Taichung, and certainly, and earlier that we have discussed, say, hey, hospital, everybody had to work together. Now I want to switch, because those tumor, the marker is the tumor tissue. But is it possible that marker come from serum? And here I want to share with you that one class of potential serum marker, which turned out to be ribonuclease. And we all ribonuclease last semester when we were young, when we studied biochemistry. This guy is very famous. 4 Nobel laureate was working on ribonuclease. Amphison folding, protein folding, using this model. Stanford Moore and William Stan to understand the function and structure, primary sequence, and then Bruce Merrifield develops peptide solid-phase synthesis at this protein. So this molecule is extremely famous in the last century. And what people know is this is the enzyme degrade RNA, and therefore it's been shown that this RNA can help cell to prevent from infection by bacteria or virus, because they can go into the bacteria inside and then degrade the RNA. That was what we know. So you may remember that in the literature, last 2-3 decades, there are people saying that there are some secret RNA, either microRNA or whatever small RNA outside the cell. And those RNA, some of them are captured by the exosome or extracellular vesicle, but some of them are free. And people suspect those RNA, some of them maybe has a growth factor activity or inhibitory activity. So just without those preliminary data, we thought, well, we're interested to identify those RNA, whether growth factor, in addition to the protein, whether RNA might be doing the same thing. Therefore, we use ribonuclease, this family, because it's very easy to commercially available. And take the cell culture and you treat the ribonuclease, and so it digests all the RNA, and to see what happened to the cell. And when we found the first, was very exciting. When you treat the RNA, well, you can test in your lab, okay? when you especially treat the riboneuclis, bovine riboneuclis, that's equivalent to human riboneuclis 5. You know what happened? We thought all the iron was degraded, but the cell growth was stimulated growth faster. So we was very excited. So we thought we may be able to identify a inhibitory factor that the RNA that degraded by riboneuclis. But our negative control is a mutation of riboneuclis, which has no enzyme activity. So the sequence is identical except single point mutation, and there's no riboneuclis enzyme activity. But that negative control still do the same thing. So i.e., riboneuclis has no enzyme activity, still stimulate cell growth. So from that point, we start to come to today's point. That the first one is riboneuclis 5. We identify with the EGF receptor ligand. This paper took a long 2 years because the cancer cell editor does not believe it. So they virtually asking us to do everything about the EGF receptor. So we did it. Second one, riboneuclis 7, then riboneuclis 1 LK, and all this was done by my lab. And then people would say, what do you mean Chi Hong? He's crazy anyway. Only his lab is doing that. But luckily, then 2022, Dr. Hu over in Boston area, it's not Harvard, what is it? Tufts University. He identified number 4 was exop ligand. So now, and here I want to say, I want to use this as an example. The reason is because RS1. And as you know, RS1 at that time is an orphan receptor. And you know, why it's orphan? And the person who cloned is right over here, right over there. Lu Hai Wang, when he was young, he was 70-something. But now when he was 40 years ago, 40 years ago? 30 years ago? When he was a Rockefeller, walking with Hanafusa, Rabbi? No, yeah, you clone the gene. Then after you clone, you just throw it there. And you never bother to identify lichen. And lichen is always, after 40 years ago, it's still orphan. But this gene is important because the rearranged RS1 causes lung cancer. And then the FDA approved drug to deal with that. So since it's an orphan receptor, so we pick it up. So hey, if we want to identify a lichen, why not identify an orphan receptor? So it turned out to be humans have certain, so we clone all of them, it's not commercially available. And we isolate all of the protein and then test it. Riboneuclis 7 binding to RS1. And here is just a standard assay to show that yes, this riboneuclis 7, number 7, binding to RS1, stimulated kinase activity and binding and form dimer and so on and so on and so on. But what's important, I'd like to share with you the liver cancer model, that we have one liver cancer cell line in which RS1 is wild type, no mutation, no rearrangement, but there's no riboneuclis 7. So that one does not really develop tumor well. However, if we transfect RSM gene in, so now the tumor becomes bigger because of oncogene addition. And then we identify this wild type riboneuclis 7 and the RS1 wild type, by RNA-seq, the stimulated gene is very similar to rearranged RS1 gene. More importantly is this. When we use an animal study and look at the serum level of riboneuclis 7, this original tumor cell, the serum level is very low, tumor is low. But once you transfect this gene in, the tumor becomes bigger, and then the serum level is higher. But now, if you use the RS1 inhibitor with FDA-approved drug, reduce, serum level reduce. So this tells us riboneuclis 7 potentially could function as a ligand and can be detected in the serum to stimulate RS1 wild-type gene, non-mutated gene. Okay, so after we came back to Taiwan, and here is lung cancer that to show that RS1 rearrangement has a FDA-approved drug. So we only have a 5-patient data. Again, no statistical significance. And as you can see here, the riboneuclis level higher, the survival rate seems to be longer. The clinical trial is still ongoing, and this was liver cancer, and we certainly are interested to expand to lung cancer since RS1 gene is rearranged in lung cancer. So, how much time do I have? I still have time? 8 minutes? Okay. So, this is another one, ribonix 1. This is ribonix 1. That one is ribonix 7. And my first one is ribonix 5. Ribonix 1, and this was as published, I believe this year, yeah. And then maybe I don't want to go to the detail, just share with you that pay attention to this one. This tumor cell, liver tumor cell, here is a macrophage. When we notice that the liver tumor cell can secrete ribonix 1 and can bind into ALK as a functional ligand receptor and stimulate ALK and go through this pathway to make the macrophage increase M2 to M1 ratio. And we know M2 ratio what? Favor tumor development. So when you have cancer cell secrete R1, then macrophage help the tumor develop. Then under this situation, if we use ALK inhibitor, which is again FDA already approved, multiple drug, and when you FDA you block ALK, then this macrophage from M2 enriched become M1 enriched to suppress tumor development. In addition, that this macrophage now secreted some cytokine to attract it. Cytotoxic T cell come in. And then when cytotoxic T cell come in, they certainly be able to suppress tumor development, for example, through PD-1, PD-L1 ligation. And therefore, this approach now allow us to use for tumor cell ribonix 1 high, we can use the combination therapy with the ALK inhibitor plus anti-PD-1, and the animal data was shown here. Without treatment, one drug, the other drug, combination. So all of this just show with you that we have identified specific marker to identify right patient population to be treated right combination drug. And on purposely, we select all the kinase because they're FDA approved drug. The drug is already available. And I have to give the credit to my boss, Dr. Ron DiPino, because when he came to MD Anderson and started the cancer moonshot concept, he keep on telling us, and I still remember, low-hanging fruit, low-hanging fruit. And he told us there are a lot of data is already there. You just need to identify the right patient to be the right drug. At that moment, we are not thinking about pitch day. We are not thinking about making money. We are thinking about a cure cancer patient. So that's why, and although the drug is already available, we just develop a marker and to identify the right patient. Okay, so the project is continuing ongoing, and we're hoping that this could become another family of a serum marker where we can identify specific patient population to be treated by existing drug. Or if the existing drug is not yet available, then certainly it will be interesting to develop the new drug, if necessary. Now, I want to come back to PD-1, PD-L1. And I remember, I believe this is very old data. I believe many people still remember that. All these anti-PD-1 or anti-PD-1 or anti-PD-L1, their clinical trial result, all these are FDA approved drugs. But they are supposed to be block either PD-L1 or PD-L1 by antibody, right? So in principle, the tumor cell should be PD-L1 positive, therefore they will respond. If PD-1 negative, then they're not supposed to respond. But if you look at the clinical trial data, this is PD-1 positive. Yeah, they all respond, very nice. So they are approved. But my friend, look at this is a long time ago, this is 10 years ago. PD-1 negative. This tumor has no PD-L1. It should not respond to the anti-PD-1 or anti-PD-L1. We still respond. Well, if I were a patient, why should I care? I respond, I'm happy. But as a scientist or as a clinician, when you see this result, you will ask yourself, why work that way? It's strange. And I hope I can convince you that these are actually false negative, just like a COVID-19 has a lot of false negative. Because if I use this as an example, this is Western, PD-L1 wild type, but once you block all the glycosylation, this is the polypeptide. So PD-L1 32 kD protein, but wild type is 50 kD, 50 plus kD. So PD-L1 polypeptide was hanging around, covered by a lot of glycosylation. And so we then come up with an idea that, by the way, both of them are still in MD Anderson, okay? Then assume this tumor cell, PD-L1 polypeptide, and all the antigen of PD-L1 to develop antibody to diagnosis are all using polypeptide. And we are the lab which first to show PD-L1 can be glycosylated, important for function and important for half-life. So assuming that tumor cell with the PD-L1, with the all different cell may have a different kind of glycosylation, but if you use an antibody try to attack this particular antigenic determined site, sometimes they can be covered by glycosylation. That was a hypothesis. But if that were the case, if we now take the immunohistostaining slide just from pathology, I'm not talking about therapy, no therapy, just a diagnosis, okay? And then those slides, if you treat the enzyme, remove glycosylation, is it possible that we could have a structure like this? And if that's the case, the PD-L1 can be easy to detect so that you will not have a false negative. And use that, we actually show that's the case, publish it, and this, at that time, we collaborate with the China Medical University in Taichung, and this patient was a lung cancer patient a long time ago. I think he was paid by himself because at that time, all these are not available yet. Before treatment, he's a CT scanning, huge tumor. After treatment, he's still alive. I don't know him, but he's hanging around in Taichung City. He's still alive. But if we took his tumor slide from pathology and just standing, it was negative. Today, the physician will tell you you're negative, you should not be treated. But then we use the same slide, use this technology to remove the glycosylation standing. I'm sorry, the positive. That's why it's response, that's why it's alive. Okay, so we have a few cases like that. And then 2 years ago, this is very interesting. Oh, 3 minutes. So no 2 years ago, this data I passed. It just tells you the same thing. Okay, so currently this project was supported by Minister of Health and Welfare, that using the Taiwan sample, that as you can see, show here, that progression disease after glycosylation, what they increase the level is limited. But all those patients, complete response, partial response, stable disease, if you remove glycosylation, their level, PD-L1 level is higher, and that's data shown here. Okay, now, here's the conclusion, but this the point I want to mention, that Taiwan need team. And I appreciate the National National Science Council, Technology Council now come up with this idea that they want entire Taiwan cancer researcher form a team. And we did form a team and apply for grant, and the grant just started this from this year. So you can see there are 7 institutions here. And this 7 institutions, in this grant, we also reserve funding to identify other institutions in Taiwan so that we can work together. So the main point I want to make is Taiwan, regardless of the field, this is the cancer field, and I heard that there's another grant from semiconductor. There are only 2 grants. But in the future, neuroscience or whatever field, that do not work individually. Do not work just on your own lab. Please work as a team, because Taiwan is very small, and our funding, no comparison. The entire Guokehui funding budget is a little bit less than MD Anderson Cancer Center funding. Okay, entire country. So we need to work together as a team. We don't complain, okay? We just need to work together, okay? And then here, earlier I showed top 10 drug, but here is top 10 pharmaceutical company. And of course, this changed from time to time, okay? And the reason why Pfizer is here probably because of the COVID-19. Okay, so all these drugs, all these companies, they are top 10, and they are all about 1 to 3 trillion NTD using Taiwan dollar. Okay, and we all know TSMC, Taijidian. This is the magic mountain that protects Taiwan, the nation, 护国神山. Okay. In 2023, it's 2.1 trillion. So, i.e., if Taiwan could develop one top 10 biopharmaceutical company, then all of us will be able to feel proud that to say, hey, biotechnology has another 护国神山, another magical mountain that protect Taiwan. Currently, we only have one, Taijidian, TSMC, right? But we need another one. And the shoulder is on what? The pressure is on what? On all of us. Of course, Wan Zhong more. Oh, of course, he has more pressure. But we all need to help him. We need to help him, okay? Well, this happened this year, right? At the noontime, we talk about AI. By the way, tomorrow, when Linda Chen talk about the lecture, she's going to mention to you the very amazing AI technology applied to cancer. Okay, so don't miss it. Okay, and then all these three turn out to be they all born in Taiwan, and they are what? Major CEO of the three different AI companies. So I don't know whether Taiwan can become an AI technology. If that happens, so be it. But biotechnology really need to catch up. So with that, so can Taiwan become a technology island? Semiconductor currently is a major mountain to protect Taiwan. And maybe AI, I don't know. Let's think about it and listen to Linda's talk tomorrow. AI, maybe something. And the other one, the biotechnology. Can we all become 护国神山? And it's all your. You should, tonight you cannot sleep, you have to think about it. Okay, that's all you need to do, okay? So with that, thank you for attention. And coming from MD Anderson, that we always like to see we work together to make cancer history and make a better life, better world. Thank you.
Okay, we have 3 minutes for question and discussion. Okay, Ron?
That was a fantastic talk. With the RNases, are they ever amplified? Is there any genomic evidence?
No, we actually checked the TCGA database to see whether ribonucleotides might actually amplify. We didn't see that.
So what regulates them? Are they transcriptional?
Most likely from the, as you pointed out earlier, that most likely from epigenetic.
I see.
Yeah.
And the mortality, the worst mortality, or let's say your point about Taiwan's mortality, cancer mortality, is not as great as it could be. I wonder how much of that is the type of cancers that the Taiwanese get. Is there more lethal cancers like lung cancer, liver cancer, that if you get it, you don't do as well, and you wouldn't do as well in the United States either, right?
That's true. That's a good point. That I don't know whether that's come from specific disease type, because the Taiwan's like lung cancer have the EGFR mutation, and liver cancer is a major disease, but not necessarily in US. I don't know the answer, but that's a good point. That's a good point. But I use that when I try to push the government to put more money in cancer. And so don't tell them. Don't tell them. Just say, hey, we have more people dying in cancer, so put more money in. Linda?
Great talk. I want to get back to the point you made about identifying patients and developing combination for the right patient at the right time. And so the data you showed there was amazing, but I want to introduce this idea that we've been thinking a lot about and want to know what you thought of it, which is, how do you think about the combination being dynamically adjusted to the patients, because each patient receiving their first drug may have very different response and different resistant levers.
Okay, so Linda just asked me a question, and then you had to listen tomorrow. She probably has the answer. Okay, so huge combination is just all known, right? So all combination, but apparently those combination therapy response is dynamic. So patient at different stage, they might be respond differently, even by the same combination, and that should come out with another level of question that we need to be aware of. And I believe your company actually is developing those AI prediction technology. I don't exactly know, but I know you're doing something like that.
We are touching on that, but actually I bring that back to our discussion at lunch, right? Because really this is why you need to connect your clinical care enterprise with your drug discovery ecosystem, because you can't keep up with the dynamic changes, the kinetic. I mean, it's a numbers game. You're fighting between what your two drugs can do versus a trillion cancer cell. And you're gonna lose if you can't keep up with them. Yeah. And the only way we can keep up is being with the patients and in real time, addressing
in real time.
And that's our next frontier.
Yes, and that we all have to keep in mind that for the therapy can be in real time, including your hospital.
Okay, time is up because we promised that he will go to high-speed rail by 2:30, okay? Let's give him a big hand.
谢谢洪校长，以及谢谢我们今日的引言人，我们陈院士，谢谢大家的参与。我们这阶段的专题演讲在此告一个段落。那待会呢，在2点50分，我们要展开今天的新药技术发表竞赛的决赛演讲部分，所以再请今天参赛的团队，麻烦在2点45分提前回到会场，来到我们会场区右侧的座位区，按照组别顺序入座。邀请大家一定要在2点50分准时的回到会场。那我们在1楼有设立茶点专区，以及我们参赛团队的竞赛海报展示区，以及本次活动赞助商的摊位区。欢迎大家可以利用休息时间多多交流，也前往参观。我们2点50准时开始，再请各位来宾朋友提前入席。谢谢大家的配合。 Hello， 现场各位的来宾朋友，我们即将在5分钟之后开启今日的决选的简报时间。今天的决选组别为新药技术组，参赛的组别麻烦请现在来到舞台区的左侧，也就是呢我们右侧的座位区，按照组别入席。稍待一会儿，我们会准时开始，再请参赛团队帮我注意时间。2点50，我们的决选竞赛会准时开始，也邀请我们想要参与的各位来宾朋友都可以自由入席。谢谢各位。欢迎我们现场所有的来宾把握时间入席。参赛团队请来到右手边的座位区，请依组别入席。想要观赏我们的竞赛说明，请麻烦帮我直接在其他座位区自由入席即可。欢迎各位来宾和我们共同参与今天的决选现场。那也请各位来宾朋友把握最后1分钟的时间入席，也欢迎大家可以集中入座。谢谢大家的配合。 Hello， 我们现场所有的贵宾，以及我们评审团队，还有我们在场的参赛团队，还有所有观礼的来宾朋友，大家下午好！欢迎大家再次的回到 Pitch Day 的活动现场。我们接下来就要进行今天的决选环节。这次呢，决选呢，首先我们登场的是新药技术发表的决选。首先感谢呢我们60多组的参赛团队，同时也恭喜所有入选团队。那我们接下来即将要展开一个非常精彩的演讲以及发表的环节。在这之前，我有请中研院生医转译研究中心陶碧华执行长为我们致辞，掌声欢迎。
谢谢今天现场的所有的贵宾，还有更重要的是这个参赛团队哦。那我们现在进到 Pitch Day 的重头戏，就是能够隆重的介绍今年在初选当中胜出的团队，来进入今天的决选。那我大概跟大家稍微讲一下举办这个 Pitch Day 活动的意义哦。那还有就是进行的大概的方式。那因为国家生技园区为了要鼓励学研界的转译团队有一些露出的机会，那特别是给这有业界经验的技术长，还有这个投资人能够参与哦，所以我们就集合的全台湾的学术团队能够共同的参与这个活动。那去年我们总共有17个学校共同参与。那今年呢就进展到20几队。那我们进行的方式哦，就是先鼓励所有的学研单位来报名参加。那今年总共有60几队报名。那我们后来呢就请每个学校出一位老师、出一位教授来去做初选的评审。那今天大家看到就是新药的10队在初选胜出的。那初选胜出之后，我们今天就是有几位呢，有非常丰富的新药开发经验的评审来帮我们做评审。那接下来我就简单的介绍几位我们今天的评审专家。好，那我们第一位哦是中央研究院生医转译中心的张正武顾问。第二位也是中研院的生医转译中心的洪伟仁顾问。第3位是全新医药生技公司的叶炳阳总经理。第4位是台湾浩鼎生技公司的赖明天研发长。第5位是顾德咨询有限公司的顾曼婷总经理。那我们这几位专家顾问，就今天的评审都有非常充分的业界的这些经验哦。那接下来呢，我想我们就将时间交给我们的胜出的转译团队跟各位评审哈，谢谢大家参与，预祝大家成功。
谢谢陶执行长，也谢谢我们今天所有的评审团队。在今天决选开始之前，由我这里先为所有决选团队报告今日的决选规则。我们今天总共有10个组别，每个组别的演讲时间共有5分钟，在你们的右侧会有荧幕计时，而同时在第4分钟时会响铃1声提醒，那在第5分钟会响铃2声。请立刻停止简报。那我们今日的决选才同问同答，所以简报完毕之后，要请决选团队回座休息。而在5组决选团队都简报完成之后，我们会请前5组的代表来到台前，各派一位来到台前进行同问同答。我们的答询方式是采1问1答，总共答询时间只有25分钟，每一题只有2分钟的作答时间。再请各位团队注意。那我们接下来呢，我们就进行今天第1组的决选团队简报。首先我们要欢迎到的编号1号国家卫生研究院暨雅慧团队上台为我们做决选简报，掌声欢迎。
好，首先我要感谢主办单位给我这个机会来介绍我们国院生药所的这个 asset DBPR 728候选发展药物 ，a kinase inhibitor targeting MYC-driven cancers。大家都知道 MYC 这个 oncogene 呢，已经被发现大概有40年，也被知道说大概有30%的癌症呢，它都具有 MYC 的基因的 overexpression 或是 amplification。那它的产生呢，如果说它有过表现或基因增幅的话，它跟这个癌症的 overall survival 呢有非常巨大的关系，尤其是在 small cell lung cancer、triple negative breast cancer 还有 neuroblastoma。具有 MYC overexpressed 的 cancer， 它的 overall survival 都降低非常的多。那我们这个 DBPR 728候选发展药的主要的 indication 呢，我们是选择 small cell lung cancer 跟 triple negative breast cancer。大概25%的 small cell lung cancer， 还有60%的 triple negative breast cancer 具有这个 MYC 基因的增幅或是过表现。那总共呢，一年在全球大概有30万人具有这样子的一个 MYC 基因增幅的 small cell lung cancer 跟 triple negative breast cancer。那除了这2个癌症之外呢，几乎所有的癌症都具有多多少少的 MYC 基因的 amplification 或 overexpression。所以呢，如果 DBPR 728可以发展到临床的话，它的整个的潜在的市场是非常巨大的。那我们所用的 mechanism of action 是利用这个 Aurora kinase 可以去 stabilize MYC 这个致癌蛋白质的这个特性。那 DBPR 728被设计说它可以去破坏这2个之间的蛋白质的一个稳定性，让这个 MYC 这个致癌蛋白质可以被细胞本身的蛋白酶体降解。其他的 Aurora kinase inhibitor 并不见得会具有这样子一个特性。另外呢 ，Aurora kinase inhibitor 也在临床上被证实说，对于 RB1 deficient 的这个 cancer 具有 synthetic lethal 的这样子的作用。那虽然 Aurora kinase inhibitor 在临床其实已经 develop 10几年了，目前是没有上市的药物。那主要的原因是因为在病人的选择上，那必须要用到正确的 Aurora kinase inhibitor， 还有必须具有正确的药动的特性。那这个就带到说，我们这个 DBPR 728的另外一个药物设计，它是一个 prodrug 前体药物的设计。它的活性药物叫做 6K465， 经由一个小小的 acylation 的这个设计，让它的口服吸收率可以高达10倍，身体利用率到达53%。其他的临床药物像 Eli Lilly 跟 Puma Biotech 的这样子，他们在肿瘤里面的浓度可以看到是绿色跟这个黑色线非常的低，而 DBPR 728的这个肿瘤暴露量是他们的10倍以上。那再来就是说，这样子的特性可以带到这个肿瘤的消退吗？可以的。那在 small cell lung cancer 里面呢，它是一个非常 aggressive 的 lung cancer， 可以看到呢，在同样的剂量下，跟 alisertib 这红色的线比，在这个虽然它们都可以造成肿瘤的消退，但是在停药以后呢 ，DBPR 728的组别，它的 cancer 呢 stay regress for more than 30 days。那在6组里面呢，有4支是 stay regress。那 alisertib 在停药之后就长起来了。那同样在3阴性乳癌的这个 Zeno 也可以看到同样的一个这样的效果。那我们做 target engagement， 看就是说到底是不是有 target 在 MYC 这样子一个特性。我们可以从这个 tumor harvest from 这个 mice， 它给1剂的 DBPR 728， 2天5天时间之后呢，去看一下 cell apoptosis， 可以看到在这个 DBPR 去组组别 cell apoptosis 非常的明显。我们也把这些 tumor 拿出来做 RNA-seq， 发现呢这个 MYC 这个 regulate 这个 Warburg effect 的一些TCA cycle 所需要一些酵素，它被 down regulate， 因而我们推断因而造成细胞凋亡的原因。所以这个技术呢，我们在2021年已经申请了 PCT 全球专利保护，在2022年呢拿到台湾专利。除了这个 efficacy 呢，还有这个 bioavailability 这个部分 ，PK 的部分，这个DBPR 728 asset， 我们也在 CMC 的部分做了非常多的琢磨。然后我们有拿到了 crystalline 稳定的 crystalline 的 form， 然后呢，我们也拿到了 clinical formulation， 所以基本上是可以马上进入这个临床转移。那这个 team 这个 work 呢是由我们生药所非常强大的 discovery team 还有 development team 所共同完成。那最后再感谢这个经费提供国家卫生研究院、卫福部、商品化中心还有国科会。好，谢谢大家。
非常谢谢来自我们国家卫生研究院的团队，辛苦了，请入席。 那么接着下来呢，就准备要来欢迎第2组团队。第2组团队呢，我们进入拣选的是来自于中央研究院旅人及赖培伦团队。那这边请来到台前，您准备好开始发声，我们就开始计时。
好，各位大家好啊，我是生医转译中心旅人的团队，那我是赖培伦。那今天呢，要跟各位来介绍我们的一个全球首创的一个革命性的细胞治疗的方法。那我们是利用小分子的药物，直接可以将人类的体细胞转换成视网膜的前驱细胞。那这样的一个技术呢，其实可以为我们未来这个视网膜这个退化的疾病患者带来一个全新的选择，一个治疗的方法。那我们的技术主要是利用这个小分子这个创新的小分子药物，在5天之内就可以把这个体细胞转换成视网膜的前驱细胞。那要跟我们的最主要的竞争对手来比呢，我们的转换效率比主要的竞争对手高达4倍，并且呢，我们可以同时去治疗黑白与彩色的这个视觉。那我们现在目前的这个跟我们竞争主要的方法呢，它是只能治疗黑白的视觉。那我们的转换效率5天，主要的竞争对手他们至少需要10天将这个细胞才可以转换成功。那另外呢，我们的这个细胞也都可以做到符合跟天然细胞一样的电生理，并且呢，也可以在动物实验里面呢恢复这个盲鼠的这个视觉的功能。那我们的这个主要的竞争对手呢，其实他们呢是还没有办法他们还没有做动物试验来证实他们的细胞功能，好，只有他们做 in vitro 的这个部分。好，那接下来就讲一下，就是说我们这个技术呢，其实它是非常具有商业价值，它的这个市场潜在收益是很大的。那目前为止，这个视网膜退化的这个疾病呢，主要有3个。第一个是黄斑部病变，第2个是糖尿病引起的视网膜病变，第3个是夜盲症。那在目前他们都是没有办法用手术的方法去做治疗。那并且呢，在这个发展到后期呢，其实基本上也只有细胞治疗是唯一的可能的最有潜力的这个治疗方法。那基因治疗通常呢都是针对在早期的，但是因为我们要在早期发现视力有问题是非常困难的一个非常困难的，所以细胞通常我们发现有问题都是在中晚期。那细胞治疗就成为一个目前非常大家被寄予希望的一个选项。那在全球呢，大概目前有200 million 的 patient， 他是黄斑部病变的这个患者，那他的潜在收益大概是11个 billion 的美金。另外，在糖尿病引起的视网膜病变，大概是9300万的患者，当然有60亿的美金。然后这个夜盲症呢，它其实是属于一个孤儿药，那它是有2亿的这个患者，大概有10亿的这个美金的这个潜在收益。所以到2030年呢，它其实到整个全球的收益大概会有200亿的美元，是相当可观的。那我们也会针对这个孤儿药呢去做我们主要的开发，并且呢去享有这个市场的这个独占期跟法规的优惠。那目前我们做了一个整个初步的统计，如果假设有10%的病患来使用这样的一个先进的细胞治疗方法呢，我们在全球的收益上面，黄斑部病变、还有这个夜盲症、还有糖尿病引起的视网膜病变，大概全球有480个 billion 的美金的潜在收益。那在台湾部分就是有4.5 billion 的美金。那我们的商业模式呢，其实会 focus 在异体跟自体的细胞治疗发展。那为什么呢？目的是希望可以提供这个医疗院所机构还有病人一个好的治疗方法。那我们也会去建构这个细胞库，然后让这个细胞库呢可以提供一个快速的运输服务，然后提供给医院端，然后去做这个细胞治疗。那在2024年到2027年，其实主要是我们的这个技术突破的关键期。那在今年2024，我们会完成 GTP cell 的 production， 另外也正在进行 pre-IND 的测试。然后到2025会进行这个 clinical batch 的 production。 然后 GLP 的独立测试。2026就会开始申请这个临床的第一期临床试验的计划。2027会得到这个人体的这个 efficacy 的 validation 的这个结果。那我们的团队很强大啊，就是由旅人团队跟这个慈济的蔡荣坤眼科医师来主持。然后我跟李佑章医师是主要的研究执行者。另外在赖倩颖小姐的部分是她跟慈济正在进行 GTP 的这个 protocol 的建设。谢谢。
感谢。感谢我们第2组的决选团队，请入席，谢谢。那我们接下来准备欢迎第3组的决选团队，是来自于国家卫生研究院谭泽华的团队。来，这里麦克风交给您。我们稍后简报更换，待会从您出声，我们就开始计时，请准备。
各位午安，大家好，我是国家卫生研究院庄怀佳，我代表谭泽华特聘研究员研究团队来向各位报告。今天要跟各位介绍一个非常美妙的 therapeutic target， 就是 MAP4K3 GOK 这个 kinase。这个 kinase 呢是谭泽华博士27年前自己 clone 出来，并且给它命名的这个 kinase。那主要呢，这个 kinase 可以作为自体免疫疾病、癌症还有 agent。inflammatory disease 的 therapeutic target。那首先我们先介绍自体免疫疾病。自体免疫疾病是慢性的，是难以治愈的，病人可能会面临死亡。但是如果病人不面临死亡，病人其实非常的可怜，非常的辛苦，因为可以有效的诊断、早期诊断或是有效的治疗自体免疫疾病，一直是医药界长期以来的一个挑战。所以病人必须终身服药，从他们罹病开始就一路服药直到他们离开这个世界。所以这也是为什么呢？自体免疫的医药市场呢，它的价值是非常的高，而且是持续的上升当中。虽然近几年的生物制剂，像是 anti-IL-17A blockade 或是 anti-TNF-alpha blockade 这些 antibody 确实可以提供相当的缓解的作用，但是呢，一来非常的昂贵，因为 protein drug 很贵。那另外一方面呢，又因为这个 treatment 呢，对于中度的或是严重度的 autoimmune disease 的病症呢，常常是没有效用的。所以其实急需更好的 therapeutic target 以及 therapeutic drug。那我们今天所介绍的这个 therapeutic target MAP4K3 GOK 呢，它是一个非常好的 biomarker and therapeutic target for autoimmune disease, cancer, aging and inflammatory disease。我们的研究团队已经证明了，在人类的自体免疫疾病里面，包含全身性红斑狼疮 （systemic lupus erythematosus, ACLE）、rheumatoid RA、rheumatoid arthritis RA， 那还有其他的 autoimmune disease 全部分列在这边。那特别的是，39% 的 ACLE patient 呢，他们都带有 GOK 这个 MAP4K3 kinase gene variant， 包含 somatic mutation 以及 germline transmitted mutation。好，那另外一方面呢？GOK overexpression 在一些 cancer cell 里面会造成 cancer metastasis。那在这边列了6种 cancer， 那包含 lung cancer， 也包含 pancreatic cancer。除此之外呢，更有趣的是，我们没有想到，我们发现在 COVID-19 patient 的 macrophage 以及 epithelial cell 里面，都过量表现了 MAP4K3 GOK 这个 kinase。而且 GOK overexpression 可以促进 SARS-CoV-2 rapid infection。好，所以这些都是我们发现的，临床检体里面就可以看到这样子的现象，而且跟GOK overexpression 与疾病的严重度高度的正相关。那我们也 identify high throughput screening identify 到小分子的抑制剂。看到 vertapofin GOK inhibitor 不像一般的 kinase inhibitor target ATP binding site， 它是 block GOK dimerization 抑制它的 kinase activity。它的 IC50 是 1.15 nanomolar。那我们发现 GOK inhibitor 放大 GOK overexpressed 的T cell specific GLK transgenic mice， 它可以明显的抑制 IL-17A cytokine。那如果在 EAE 或 CIA mouse model 里面，也可以显著抑制疾病的严重度。而且很特别的一件事情是 ，GLK inhibitor 可以 induce regulatory T cell Treg 的 differentiation。那在 human T cell 里面一样可以抑制 IL-17A 的 production， 并且抑制 AHR gamma T 这个 complex， 这是可以 induce IL-17A production 的 complex。那这些是 cancer model 以及 SARS-CoV-2 model 也都有一样的效果。那特别是 GLK knockout mice 会长寿，而且没有什么疾病，特别是它毛发非常的黑，非常的漂亮，可见 GLK inhibition 不会造成什么特殊的 effect。那这边有个很特别的事情是 ，GLK induce IL-17， 而且它抑制 Treg， 反过来抑制 GLK 会 inhibit IL-17A， 而且 induce Treg。这就是为什么抑制 GLK 会比抑制 IL-17A 而已会更有效，而且它可以作为一个选择病人的 biomarker。
麻烦请停止简报，谢谢。感谢我们第3组的团队，来自国家卫生研究院，辛苦了。那我们接下来呢，要邀请的是今日第4组的台语生医股份有限公司林炳熙团队为我们进行简报。这里我们做一下简报更换。若您准备好开始发声，我们即开始计时。
大家好，我是国家卫生研究院李敏熙研究员。经过30年疫苗研发的历练，我最近带领团队在国科会的补助下成立新创公司台语生医，将利用基因工程技术开发次世代肠病毒及流感疫苗，能够快速开发符合成本效益、更安全有效的疫苗。肠病毒疫苗第1代的疫苗是利用野生病毒株来生产，产能低，只有针对 EV71 型有保护力。我们正在开发的第2代疫苗是将利用基因工程开发高成长的 EV71及克沙奇16的高成长病毒株，产能高且具有广效保护力。那在流感疫苗的部分，第1代的流感疫苗是利用鸡胚胎生产。虽然便宜，但是它市占率约85%，但是有3个主要的缺点。第一个，准备期长，大概需要20周，需要生产活病毒，有生安疑虑，容易基因突变，降低疫苗保护力。第2代的流感疫苗是用哺乳类细胞生产的不活化疫苗，除了上面提到3个缺点，它只能改善其中一个，减少突变的问题，但市占率约10%。第3代的疫苗是利用昆虫细胞生产的重组蛋白次单位疫苗，它可以把上面3个缺点都克服，同时也可以因应新兴传染病来快速开发疫苗。但是它有一个缺点，致免疫力比较差，比较昂贵。目前的市占率大约只有5%。我们正在开发的第4代疫苗是利用昆虫细胞生产的重组蛋白类病毒颗粒疫苗，它可克服上面3个的缺点，同时致免疫力比次单位疫苗好，生产成本也比次单位低，具有更好的竞争力。在市场规模方面，肠病毒疫苗主要是亚洲市场，中国每年使用大概1600万剂疫苗，每剂大概30块美金，市值约5亿美金。在台湾去年上市使用了大约20万剂疫苗，每剂大概140块美金，市值约3000万美金。东南亚主要市场是有越南、马来西亚、泰国。泰国今年上市了，每剂大概100块美金。预估市场的亚洲市场、东南亚市场应该有200万剂，总共大概2亿美元。在流感疫苗部分，季节性流感疫苗在2023年的使用率大概有80亿美元，每年成长率大概8%，预估到2030年大概有140亿美元。如果发生新型流感大流行，市值大概产值约超过100亿美金。台语生医聚焦在研发及早期临床试验，将与亚洲的疫苗厂合作开发肠病毒疫苗，与跨国的疫苗厂开发流感的疫苗目前都有团队在洽谈中。 在竞争者方面，肠病毒多价的疫苗，目前有3家亚洲公司进入到第1期临床试验，台语将于明年第4季申请 IND， 所以跟国外的整个大家的期程差距不大。在重组蛋白疫苗部分，赛诺威是唯一有一个次单位疫苗上市的公司。那 Novavax 现在正在进到第3期临床试验。那在 Nbrp 疫苗部分，目前是只有台语生医将在近期启动临床试验。我们预估在2024年募资2到300万美金，逐步完成临床试验以后，完成第1期临床试验，预估在2027年申请 IPO。目前是 CDE 的指标案例。我们在国院建立完整的研发团队，还有完整的 IP 布局，也建立了坚强的研发顾问。台语生医立足台湾，放眼宇宙，是疫苗开发的好伙伴。谢谢大家。
感谢第4组的团队为我们进行的简报，谢谢。那接下来我们欢迎第5组来自台北医学大学潘秀玲团队为我们进行简报。
大家好，我是宋婷怡。那我代表台北医学大学潘秀玲院长团队来跟大家介绍一个我们应用在临床前小分子新药开发的一个 AI 的药物筛选跟设计的平台。那以往呢，我们就是需要花上2到3年的时间才可以去找到一个有效的 lead compound。可是我们团队透过了这个 AI 的平台，我们在3年内已经找到超过70个有效的 novel 的 lead compound， 甚至是 optimized 的 lead compound。那这样子大幅的就是加快了我们研发的速度。那今天很开心跟各位来分享我们这个 AI 的平台。我们预计在今年10月的时候会成立衍生新创公司，来正式提供服务给所有新药开发相关的单位，那提供产品跟服务内容。大家看到左边这张趋势图，浅绿色的部分是统计过去10年来全球20大药厂他们所拥有的临床前新药研发专案的数量。那可以看到，2021年相比于2010年，它的研发专案的数量大幅缩减了53%，这是代表着新药研发是越来越困难的。经过统计，平均药厂要在研发一个临床前的新药的话，它必须要花费12亿美金以上，甚至是5.5年以上的时间，来去得到一个可以进到临床试验的新药。而我们的 AI 平台可以以更少的经费、更快的时间，去让我们获得一个有效的lead compound。我们的 AI 平台在一开始设计的时候就已经囊括进 AI、化学合成还有生物药独立的团队。那以团队这样子一个3方共同的合作之下，我们大幅的提升了我们 AI 平台在设计新颖药物的一个可用性。我们的服务范畴呢，就包含了除了大家常常听到 AI 公司会去做的compound screening 啊、structure optimization 以及 tax prediction 等等以外，我们的 AI 平台更着重在我们 AI 所设计出来的新颖的 compound， 它必须要是可以用的，它是有效的，它是可以合成的。除此之外，我们也建立了 protein target screening model， 再搭配了就是生物药独立实验的验证。我们 AI 平台主要的优势有3个，首先，我们的平台可以去设计出具有可专利性的新颖结构，那这些新颖结构呢，还有80%的可合成性。那这些可合成的结构里面的，在生物药独立实验的验证下，它也具有非常高的有效性。在设计的就是新药新颖结构的部分呢，我们 AI 平台就涵盖了1300个以上的 protein target model。除此之外，我们自己也建立了一个 virtual compound library， 里面涵盖了3000万笔的 virtual compound 的 data。那由此，我们的 AI 所设计出来的 compound 结构基本上是具有95%以上的 novelty， 同时具备很高的这个可合成性。那由于就是我们 AI 的设计的结果，我们可以降低我们的就是 compound 的90% 的合成的成本，并且缩短合成时间到3到7天的时间。 这些 compound 进到细胞实验之后，有50% 的数量它可以 IC50 表现小于10 micromolar。那我们团队就依照这样子，以这样子一个很大的优势，在3年内就开发出77个有效的一个新颖的 lead compound。那这些 lead compound 都是 IC50 小于10 nanomolar。那这些 compound 里面就包含了29个 protein targets。我们走的最远的几个研发专案已经涵盖到了 preclinical PK study 的验证。我们在10月成立公司之后，主要会提供3个服务项目。第一个是早期新药的一个早期新药产品的授权，那包含了 cancer 还有 neurodegenerative disease 的一个 indication。第二个部分是 AI 在临床前新药研发的一个 R&D services。除此之外，我们也可以跟客户一起共同开发新药产品或者是新的 AI model。我们就是截至目前为止呢，已经服务了19个客户，那这19个客户包含了9间生技药厂、6个学研单位，还有4个法人研发单位。我们的营收也是逐年成长。那很高兴跟大家分享一个好消息，就是我们今年10月之后会承接一个日本大药厂的一个委托案，为公司带来6到7万美元的盈利。那随着未来我们一直逐渐扩张我们的客户服务量能，我们预计在2027年的时候达到60万美元的营收。那以上非常感谢大家的聆听，谢谢。
感谢第5组来自台北医学大学的团队，那也请可以来到台前进行 Q&A 准备。我们接下来呢会进行1到5组的 Q&A 时间。那邀请呢第1组到第5组的代表，请派1位来到台前，请依序由面对评审的方向，由左至右来进行入座。请各位代表上前。那和各位评审报告，我们第1组是在您们的右手边。好的，那呢，再次的为大家说明一下 Q&A 的规则。我们 Q&A 时间总共是25分钟，采评审1问1答，每一题只有2分钟的答询时间。我们也会按铃提醒，答题1分钟会按1下，2分钟会按2下。请立刻停止作答。那要作答前，请记得要先拿起我们桌前的麦克风。那接下来呢，我们就让评审从您开始发声，我们就开始计时。
先问第1组雅慧，这个 MYC 的 inhibitor， 我晓得是 Aurora， 所以你也跟 Aurora 在去年8月份 approve Puma 的 alisertib。当然 ，Puma 这个 compound 是从 Millennium 买来的，因为 Aurora 就是毒性偏大，最后在 small cell lung cancer 得到成功。刚才看到你的就是老鼠的 data， 你跟它的 potency 是差不多类似的哈，可是你的 dose 是非常非常的大，100 milligram 啊。那请问你，因为它在8月份已经上市了，对，它是8月2023 FDA approved orphan drug 呀，那所以你跟它来比较有什么 advantage？ 你觉得5年之后你上市的话，可以就是说抢到它的市场吗？
好，那回答顾董，那基本上 Aurora 这个 alisertib 它是在去年8月是得到 FDA Phase 2 approved 的 orphan drug 的 study design， 他们现在还在 recruit patient。那我认为就是说 alisertib 它应该在这个study design， 我认为它会成功，因为它在我们的 animal model 里面也得到非常好的 tumor regression。但是问题是 small cell lung cancer 也可以看到他们的 data 里面，在 drug treat 完之后，它会马上就是 rebound。主要就是因为他们对于这个MYC 的这个 suppression 并没有像我们的药物的这么好。那其实，在过去的 literature 一直想要用 alisertib 去看到 MYC 在 tumor 里面的 down regulation， 但是他们都没有看到，有可能是因为他们在 tumor 的 penetration 没有那么好。那因为他们需要 BID， 就是他一天要吃2剂，但是他在 tumor 里面停留时间就只有8小时。但是问题是因为这些tumor 这个 MYC amplified tumor， 这 MYC 这个基因它其实是 它的 half life 只有20到30分钟，所以如果说它在肿瘤里面的药物浓度不能维持的话，它 tumor 就会 rebound 回来。这也反映在我们在 xenograft 的 model 里面看到，他们 drug 停药之后马上就 regret， 但是因为我们的药物的 tumor penetration 非常好，而且它 half life 很长，所以我们认为它在有效浓度一直维持的状态下，才有可能造成有效的 tumor的 regression。那我们认为我们的效果会比它好。当然，他们是一个好的，我觉得药物的开发就是这样，它又一代一代。如果有发展比较更好的药物，能够在临床上得到证实的话，那它其实有前面的 study design 是一个好的一个可以学习的地方。谢谢。
我不知道我可不可以问 follow up 的问题哈，也是第一组。您刚刚讲到，就是在您的 presentation 里面，其实就是 went through very fast about 这个 prodrug。我不知道 prodrug 它的这个 cleavage 它的是不是 species dependent， 因为我没有 catch 到你的那个 mechanism， 是 chemical induced 还是 enzyme induced？ 那您刚才提到说 half life 很长，那它这个 half life 是 in the intact prodrug form 还是 free drug 的 form？
好，那关于这个问题呢，其实我们也有 study 过。那其实它不是，因为它是用一个 acylation， 说它是一个 hydration based、pH dependent， 当然它也有可能 enzymatic， 所以在不同 tissue 里面，它的确有不同的 conversion rate。那我们有看过，它在血液里面代谢是变成 active compound 是比较快的 ，clearance 也比较快。但在 tumor 里面呢，它 conversion 就是说从肠胃道一直到 tumor 里面，它在 prodrug 这个形式的保留是比较长的，那当然它会 slow release 到它的 active compound， 所以 overall 如果是在高浓度剂量下，它的有效浓度可以维持到5天左右，但是如果在低剂量下，当然是不见得是这样子。那所以就是说它有没有 species dependent？ 我们有做过狗、大鼠、小鼠，那其实它都可以 convert， 因为它是一个 base 很 simple 的 chemistry hydration。还有 enzymatic， 当然是有 tissue dependent。那之后的这个我觉得在 GLP tox 的时候，可以再更详细的去 investigate 这个问题。谢谢。
我想问一下那个吕仁跟赖培文这个presentation， 那因为你这个是small molecule， 然后是所以我想了解一下，就是说你在开发过程，那个有没有考虑到，就是说它这个 drug interaction， 它的整个这个PK profile， 还有它 metabolism 的 pathway， 也就是说在你在开发的过程，你有没有一个 idea 的 target product profile？ 就是说我们在开发药物的过程都要有一个，就是说你要开发这个药，你本身想要这个药具有什么样的特性？你有没有在市场上有没有竞争性？所以这方面你们有没有考虑到？
谢谢委员的问题啊。那我们其实主要是着重在细胞治疗的方面，所以我们的新颖药物其实是细胞本身。那在做成这个细胞的过程里面，其实我们有用了一些已知的跟一些 FDA 已经通过的小分子药物在做这个细胞的诱导。但是至于就是说，在对于这些小分子药物本身呢，我们就比较没有去琢磨，因为其实我们的团队其实是干细胞学家的团队，所以在于药物上面的话，我们那时候的认知跟一些 background 是对于就是这个跟药理跟药物专家比起来，我们是比较还没有到他们的这么的精准跟专业。但是在细胞上面，就是我们其实之前有被问到一个问题，就是说当这个小分子药物诱导这个细胞变成我们要的 target cell 的时候，它是不是在临床在使用的时候，是不是可以被清除干净、被洗干净？这个是有被问到的一些安全性的问题。那我们现在其实也正在努力的建立那个 GTP 的这个 protocol， 看是不是可以符合就是法规上面的要求跟标准。那所以就是希望我们这个细胞的产品，它其实是可以顺利的符合这些再生医疗目前推广的这些法规。对。
那我想我也是有一个 follow up 的问题，同一组哈。您刚才讲到说，你们这个 cell 的这个细胞治疗的方式是 for late stage，right？ 那我的问题是说，你们会 expect 你们的 treatment 跟现在既有的 treatment 你们会是 the first line、second line 还是 third line？ 我随便就是问一下，就是说你们的 market entry 的计划，因为前面一定会有其他的 treatment follow up， 所以也许请您赘述一下，谢谢。
谢谢委员的问题。我们其实是比较针对中晚期，因为基本上我们的这个发明人主要是眼科医师哦。那据眼科医师他们的这个医学背景来讲的话，可以发现到这个视网膜退化性疾病的产生呢，在早期的时候都是没有办法顺利的被发现，然后通常有时候会觉得是可能是太累啊或者什么的，它所引起的。所以在 genetic 的上面的话，是基因治疗，它大概都是有基因型的限制，就是说如果你很早就可以去做 genotype。typing 可以知道说是 RPE65的这个 mutation， 然后它就会造成这样的一个视网膜退化，但很少就是这些基因治疗的话，它是针对于部分的这些病人，他有办法去做治疗。可是细胞治疗就不一样，是当你这个细胞死光了以后，你可以再重新注入这些人造的细胞进去 rescue。它的这个 vision 就是它所 lost 的这个 vision， 所以目前来讲的话，细胞治疗其实是比基因治疗还有一些特定的就是回转性的药物，像一些叶黄素这一些的营养的这一些补充品，还要来更有希望的。
question 第5对哈，就是用 AI generative small molecule。我有注意到你说你的 validation。在 cell-based study 是 IC50 小于10个 nanomolar， 我不晓得你知不知道，小于10个 nanomolar 是非常不 potent 的一个情况哈。一般我们 generative AI 的 target 是 molecular engagement 是10个 nanomolar， 那跟你的 target 是相差了1000倍，那不晓得。你们团队对这样的事情有怎么样的想法？谢谢。
谢谢。那跟委员介绍了一下，就是我们现在呢，就是主要会 AI 插在就是设计出来的结构之后，我们合成后，我们首先会经过 kinase assay 去测定它的最初的 IC50， 这个时候的 criteria 是10 nanomolar。那等到进到细胞实验的时候，我们去筛检的那个就是 criteria 会是10 micromolar。那当然同时间，我们目前也有就是将近10个的 compound 是 IC50 小于1 micromolar。那其实我们也正在努力去降低这个就是等级。那目前也有获得一些 IC50 是大概0.几 micromolar 的对 compound。那因此说我们正在持续努力的提高它的 potential。谢谢您。
我想请教一下吕人老师这个团队哦，就是你用 cell therapy 去治疗这个失明。那现在有另外一个方法是用这个 chip implantation。那之前有比如说刘文泰刘教授，那现在 Neuralink 也 claim 说他们也打算往这个方向去做，也有一些 preliminary 的 evidence。那你怎么去比较说你的 cell therapy 跟这个 chip implantation， 那它的这个 efficacy 跟 cost benefit？ 谢谢。
委员谢谢。委员您提到就是说就是在眼部植入晶片这个，那我们之前其实有去研究有一些在美国他们已经 FDA approved 的上市的那个它的人造电子那个视网膜晶片，那其实他们的效果其实老实说我们去研究了一下，他们效果并不好。那他们在临床1期的时候 OK， 然后在2期的时候就宣告他们的效果差，然后就下架了这样子。所以其实目前就我们团队所知道的，就是在晶片上面的移植到视网膜上面，其实它的效果是不好的。那刚刚因为我们有提到，就是说我们未来产品在定价上面的考量是1眼20万美金，然后其实也是参考了这个晶片移植跟另外基因治疗的药物，我们就是取它的在第1眼的价格这样子。所以目前我们是认为说在晶片上面它的效果没有我们的细胞治疗来的有潜力。
我有个问题，想要问我们第3组 谭老师这边，谢谢你们介绍这个 GOK 各式各样的机转可以用在很多的适应症上面。It's a good news, but it's a bad news too， 因为有太多的机会，那你们如果 moving forward 有资源继续往下走的话，哪一个适应症会对你们来讲是最有兴趣的
？OK， 那个今天之前的 talk 也有讲，就是说事实上我们可以从一个 indication work 以后再推到下一个。那我们长期以来一直是说 autoimmune disease 为 target， 所以那个是我们以最多 animal model 最多证实的。Aging 应该是最这是很非常有前瞻性，可是事实上我们没有多少 data。我的 data 就2个，一个是老鼠可以活很长到3年5个月，然后另外一个就是他们的那个，我们认为它就是因为 information 下降，所以是相关的。所有这些都是 information。那最有相关的应该就是autoimmune， 所以这个是我们会。
即便是 autoimmune 也有好多适应症。
对，
哪一个你会认为胜算最高？
我们一开始我们目标定得很高，很多人是觉得不实际。我们是定 lupus 因为最难，到现在为什么？因为没有可以，就是还没有好的药。那但是如果要实际来讲，我们可能会从 psoriasis 开始。因为 psoriasis 也是非常 sensitive to IL-17 blockade 的 indication， 所以我们可能会从那边开始，然后但是我们目标也希望到解决到真的没有药的这个 disease， 这是我们希望能做。那当然有其他的我们的 along the way， 但是一定是 IL-17 mediated 的 disease， 或者是 IL-17 blockade work 的 disease。这个我想这是给我们一个好的，但是就是不是大概是僵直性脊椎炎或是 psoriasis。
我想问一下第5位的 presenter， 就是说目前 AI 用在各种领域的应用很广。那这样我想了解一下，就是说你们 AI compared to others，what kind of comparative advantage？ 你们优势在哪里？那你们用的这个1400个这个蛋白的 target， 他们都有 X-ray structure available, or what would be the database you need to design a good drug? So first, what kind of advantage you have compared to others?
OK， 好，那谢谢委员的提问。那我们的 AI 平台，它主要跟其他 competitor， 经过我们有去接触过很多国外的一些 AI 的型号设计公司，包含也有国内的也有接触过。那我们发现，其实我们的优势就是在我们的可合成性，因为很多的 AI 它目前面临到的问题，就是它设计出来的新药结构是难以合成的。那所以他们就需要另外一个 AI model 是去 predict 它的合成的途径。可是当这样子的情况发生的时候，合成途径它的结构，它可能如果本来是难以合成，它就算 predict 出来了，它最后要去花费在合成上面的经费跟时间也是很广的、很大的，那也会影响到它后面的 scale up。所以我们就在这个合成的这个部分呢，我们一开始的时候就跟化学合成的专家去做一个很密切的讨论，说，我们 AI model 应该要在哪个地方去modify， 然后去 optimize， 那得到的一个结果就是让我们的 AI 的产生的一个 compound 的结构是容易可以合成的，而且它必须要在细胞实验上面是很容易有效性，这样才可以往下推展到动物实验，再进到 preclinical。那您的第2个问题是关于到我们的 protein target model。那我们比较特别的是说，我们的因为一般来说 protein target model 他们可能去建立这个 model 的时候，他们的资料是全部破在一起去训练的。那我们是针对把每一个 protein target 都分开来，然后收集他们相对应的 compound 资料、结构资料，然后去 training。所以每一个 protein target 它都会有各自独立的 library。那经由这样子，它会提高这个就是 prediction 或是设计的一个专一性跟就是一个精准度。谢谢您。
我可以 follow up 再问一下。
没问题。
您恭喜啊，马上要成立新公司。
是。
可是江湖上不管在台湾，在世界各地有很多的 AI 的 R&D 的公司在做你们类似的事情。 那你说到有些公司已经跟你们谈，有可能会拿到一些 project。How do you differentiate yourself？ 他们你怎么说？什么事情是我们做得到，别人做不到的呢？因为这竞争实在太激烈了。
对的，那就是光是在 market differentiation 上面，我们其实就有做一个区隔，因为像 USA 跟 Europe 那边其实竞争是非常激烈，可是，在就是 APEC 这一区，其实还算是一个还在萌芽、正在成长的一个东西。那我们刚刚就是前一位委员提到的问题，那我刚刚延续提到说，就是我们其实接触过其他的 AI 新药公司，他们其实很多一开始的时候，他们就只有 AI team， 或者是可能单独1、2个。chemical 的，或者是就是等等相关的人员很少，所以他们是 AI team 单独打斗起来建立这个 model。那其实这样子的建立起来的 model， 它在升级新药上面的话，就会有一些没有办法去设计出可以合成的或者有效性，因为他们缺少了一些 critical 实务上面的经验跟知识。那其实这就是我们最大的一个不同的地方，因为我们 teamwork 是非常的紧密的，我们一开始就有 AI 跟化学合成，还有生物药毒理实验，然后甚至是我们团队都拥有3个 IND 的经验。那因此在这些经验的累积下，会塑造出我们的 AI model 在本质上跟别人不同。谢谢。
第3个团队哈，我觉得那个 GLK Discovery 是非常了不起的一件事情哈。那因为这个东西在1997年哈就已经有了，到目前为止的话，好像没有引起大药厂的兴趣哈。那 IL-17当然是非常的重要嘛。那 IL-17的药已经都出来有一阵子了哈，也是非常的有效。那请问哲华，就是为什么大药厂缺乏对 GLK 的兴趣？那它是 IL-17的上游，它的 pro and con 跟 IL-17直接的 inhibitor 比较有什么不同？
好，我先回答这个第二个问题，就是说它跟 IL-17 blockade 的效果有什么不同？就是 pros and con。那我想第一个最主要，当然那个是 antibody 药，我们这是小分子药物。我 I tend to like the small molecule inhibitor， 因为最后是比较可以推广的，我们就是比较大家都可以用到的。然后这是一个。那第二个呢，它是明显的，在我们的实验 validate 呢，它是明显比 IL-17 blockade 还要好，因为它还会因为 GLK 还会这个抑制Treg regulatory T cell， 它是 inhibit T cell。那所以 inhibitor 的话呢，它就会加强 Treg， 所以它除了 IL-17 blocking 那个效果以外，它还可以会 inhibit 不就 induce Treg， 所以呢，它可以再更强效的再去抑制这个所有 T cell 的那个 activation。所以我想这是一个最主要，这个我们找到这也是非常 surprise， 这是后来最近1、2年后来才看到的。一开始的话，我们只认为只是 cost 的问题。那另外一个为什么这么好？为什么没有大公司做？那我要讲，你看我之前在我们发现的也是我们 discover 的更早，是96年，在还要早28年前，叫 HPK1， 它现在已经是在那个 cancer immunotherapy 已经是 hard kinase 了，所以呢是同样一个 family member 的，那它是反过来是 inhibit T cell， 那为什么没有呢？我想第一个呢，这个 MAP4K 这个 family 它很大 ，protein 很容易被degrade，所以一般人在实验室他们不太容易去看到这个 protein。那我们有 know how， 所以我们可以看到。我想这是一个最主要的，大家没有办法跟上，所以很多人这次去看了以后，他没有办法去验证它到底好还是不好。我想这个可能是大家最主要的 problem。
请问是否还有评审需要提问？若没有的话，我们就提前结束 Q&A 环节。谢谢各位评审，也谢谢所有参赛决选者。谢谢，请入席。 接着下来我们会休息5分钟的时间，我们会在呢3点55分前再次的开始下半阶段的决选。辛苦评审，也谢谢现场所有来宾的参与。那下半阶段6号到10号的决选组别，请依旧来到我们会场右侧的座位区小左等候。我们5分钟之后马上回到现场，谢谢大家。 Hello， 现场各位贵宾，请尽速回到座位区，也邀请评审帮我再次的回到评审席入座。我们下半阶段的决选竞赛简报即将开始，谢谢大家的配合。Hello， 现场各位贵宾，欢迎再次回到 Pitch Day 新药决选组的竞赛现场。那我们接下来呢，就进行6到10号的团队简报。再次提醒，今天简报规则，每组有5分钟的时间，4分钟响铃1次，5分钟响铃2次，请立刻停止简报。我们也一样会在最后进行统问统答，再请各位团队注意。那我们接下来呢，就有请今天第6组的团队硕准生技股份有限公司吕运琪团队来进行简报，请上台。请您准备好，开始简报。我们出声即开始计时。
各位前辈先进，大家好，我是硕准生技的运琪，今天很荣幸能跟各位报告硕准。硕准的核心目标就是我们希望开发利用我们的抗体所，可以让所有的药物精准的作用在靶点，让抗体药物可以有更好的疗效、更低的副作用，提高给病人更好的生活品质。为什么我们会想要做这个题目呢？我们可以从刚刚洪明奇院士的分享中知道，生技药物它在现在目前有很大的市场，但是说抗体药物也并非十全十美，所以有时候抗体药物打进去人体内以后，不只作用在肿瘤区域，无差别的综合所有的抗原的时候，就会带来副作用。我们可以看到大肠癌的药物 Erbitaz 为例，在EGFR 这个靶点在皮肤上面也有过度的表现，就导致了皮肤毒性。而当现在的 scientists 为了想要提高药物的疗效，开发成 ADC 的时候，也因为了 ADC 有更好的疗效、更高的毒性，导致像 AV Immunogen 在临床试验中就因为皮肤毒性，整个 trial 就终止了。所以我们团队有一个非常简单的概念，我们利用抗体的腰部的 hinge 形成一个空间性的结构，并且使用 protease 这个 linker 作为连接，形成一个抗体锁。它就可以让抗体药物在正常组织的时候没有任何的功能，只有到达疾病区域的时候，会有过度表现的一些 disease associated 专一性的蛋白酶，它才可以进而把我们的抗体所解锁，移除以后，那这个抗体药物才可以精准的作用在疾病区域。我们目前已经利用这个抗体锁的技术，成功的应用在10种的抗癌症药物、3种的自体免疫疾病药物，确认它可以提高更高的安全性、更低的副作用、更好的疗效，以及它可以应用在任何的抗体药物上面。目前呢，我们还有2个 super tool， 一个是来自于整个台湾检体资料库，超过临床500个癌症病人的检体，让我们知道什么样的蛋白酶最适合作为解锁工具，并且利用我们的电脑模拟系统，我们可以预测什么样的抗体锁它可以有最好的屏蔽能力、最高的活化几率，让我们不止挡得好，也要活化得好，有好的安全性，也要有好的疗效。那目前呢，这个抗体锁我们也针对了抗体药物所有全球的市场，布局了18个国家，也全数的获证了。那平台技术最大的好处就是我们每锁一个药物，就是一个新的组合物，一个新的专利。 那目前呢，我们第一个产品 PSA101也预计在9月的时候，这个月我们是已经要准备 filing provision 美国专利了。那我们的与竞争者的最大的比较就是我们是来自于抗体自身的腰部作为屏蔽区，所以我们有最低的 immunogenicity。那有关于解锁效率呢？先前其他公司的竞争者药物，他们大部分在2期的时候就临床失败了，疗效不彰。那我们进而发现他们的临床解锁效率只有20%，所以透过我们的 biobank 来自于500个种的简体的话，我们可以有最好的解锁效率。那其他的竞争者呢，分别像 C 菌跟 Cytomax 以及 C6， 他们分别都是美国上市公司，也有重大的产值。那目前呢，我们首要的 lead compound PSM151 已经证实它可以降低皮肤毒性40倍。那利用 Chrome Bio 中美矿科的 PDX model， 我们也验证了它有更好的疗效。也见证了它跟现在目前的 Cetuximab 有一样相似好的疗效。那目前呢，我们也有跟全球的其他药厂进行negotiate，未来我们在完成50例以及猴子的副作用测试的时候，有机会早期 license out。那我们这个药物呢，第一个我们会攻进头颈癌这个比较少竞争者的药物市场。再接下来呢，这个 EGFR 药物还可以到肺癌以及大肠癌，而我们的抗体锁呢，还可以在各项的像单抗、双抗以及 ADC 都可以适用。我们可以借由 license 有一部分的 revenue。那目前我们除了单抗、ADC 以及 autoimmune disease 都是我们目标范围之一。那我们的抗体锁有3大应用范围，针对专利过期的药物，我们透过抗体锁可以让专利新生；针对副作用过强的药物，我们可以让它复活；而没有办法打开的市场，我们可以让它 ADC 产生更大的市场。那我们就是透过抗体锁的服务，目前也已经提供这样抗体锁服务给一间厂商，并且进而在今年5月的时候，我们很荣幸的授权我们的技术给台湾唐联。那未来随着我们的新药发展，我们可以水涨船高有关我们的技术。谢谢。
辛苦了，来自我们第6组的团队，请入席。接下来我们要欢迎第7组的团队，是来自于国立成功大学庄伟哲以及张耀宗团队进行简报。那我们这里呢，更换一下简报荧幕，那请您测试一下麦克风以及简报笔。若您准备好，我们即开始计时，谢谢。
好，各位委员，然后以及各位先进，大家好。那我们是来自国立成功大学庄伟哲教授的团队。那我们最主要的一个发明呢，在这部分我们就是发明了一个低出血风险的，然后针对血小板上阿法图伊贝塔3组合蛋白的一个溶栓剂。那我们都知道，目前心血管疾病事实上还是10大首位的一个10大死因首位的一个疾病。那目前的药物治疗呢，事实上都会面临一个很严重的一个问题，就是出血的问题。那超过10% 都会面临不管什么药物都会有这一个出血的问题。那而且我们都知道，出血往往都跟所谓的致死率有一定的正相关。那所以造成出血之后呢，我们接下来的用药的指引就是一个判断出血一个非常重要的一个东西。那只要有判断出血，我们一定就是进行一个所谓的没有一个药物治疗使用的情况底下，那这样的情况底下呢，会造成的有可能是一个闭锁性的一个栓塞又会出现。那当然我们的出血的反应就会被下降下来。但是呢，我们在没有判断一个出血的情况底下呢，我们持续的一个治疗药物，我们发生什么情况呢？虽然血栓可以被抑制得很好，能够被抑制得下来，但是在我们的凝血的这一块呢，一样是被抑制的，所以一样会造成持续的出血，以及造成更大的副作用。那依据目前的了解呢，就是我们可以针对一个所谓的奥赛因的一个抑制剂呢，来达到一个所谓的一个筛选的一个抑制效果。这样的一个抑制剂呢，能够有效的抑制闭锁性的血栓，但是呢，不会抑制初级的凝血。那这也是从文献里面我们所得到的，其中一个关键的一个技术呢，就是在血小板上的 integrin alpha 2 beta 3。我们都知道 integrin alpha 2 beta 3在受到我们的抑制剂活化之后呢，它会被所谓的人体的一个抗体做辨识，然后进一步造成 integrin 的活化，然后做清除。也就造成出血的一个原因。那所以我们其中一个筛选的方式呢，就是开发出一个抑制剂呢，它不会去活化 integrin alpha 2 beta 3。那另外一个呢，就是针对就是所谓的初级凝血这一块呢，我们透过 cell 的移循的方式呢，来做一个筛选。 我们透过了超过200种以上的突变蛋白的设计以及筛选呢，我们能够达到一个高安全性的一个蛋白，就是 RR 这个蛋白，它能够有效的抑制的所谓闭锁性的一个血栓，但是不会抑制初级的凝血，也就是不会造成出血的副作用。那所以除了在小型老鼠的动物实验验证之外，我们更在大型动物里面已经验证到这样一个安全性。那我们在不管是在血小板的一个数量，以及在出血时间上呢，我们跟目前市售的一个药物做比较呢，我们的药物呢并不会影响血小板的数量，不会导致它做血小板的清除。此外呢，出血就是一个很好的一个 indicator。我们的出血时间呢跟市售药物比呢也是不会被延长的。那在疾病的 model 里面呢，我们使用的是氯化铁所诱导的一个肺动脉的栓塞的这个实验。那相同的与市售药物做比较呢，我们的药物呢高安全性这 RR 的这个蛋白呢，除了能够有效的抑制这一个闭锁性的血栓之外呢，我们更有一个隐藏性的一个效果，我们可以避免血管的再窄化。我们都知道血管再窄化可能是进一步引发你的再栓塞以及一个复发的一个作用的另外一个潜在的问题。那此外呢，我们也开发出另外一个蛮有趣的，就是从肌肉注射的方式来达到治疗的一个效果。那我们都知道，目前的血液治疗除了所谓的血管注射，那另外就是口服。那我们开发的是另外一种选项，就是我们可以在不管是在居家或是在紧急治疗上呢，都有可能会有未来的治疗潜力。那此外，我们进一步与目前市售上的柔血酶呢做 fusion 蛋白，做成融合蛋白，我们也验证了出来，它具有一个比较好的一个治疗的区间，在高浓度底下。那我们目前呢，不管是在单体的这个 RR 蛋白以及融合蛋白的这个专利布局上呢，都已经有进行布局。那我们朝向的就是说，我们可以先把我们的这个单体的部分呢，如果找到一个合适的厂商可以进行技转，接着呢，我们也会持续进行我们开发我们的融合蛋白的2代药物以及未来的 BioBeta 的一个开发。那我们知道这市场事实上是很大的，那年成长率大概有超过9%。那我们的 indication 呢是朝向目前的一个治疗方式，像是 non-stemming 或是 angina。那我们的团队就是庄伟哲教授，那感谢我们这些团队的一个支持合作以及我们的合作伙伴，谢谢大家。
感谢我们第7组的简报，谢谢您，请入席。接着上来，我们欢迎第8组来自中国医药大学黄伟谦团队为我们进行简报。荧幕上我们更新一下简报画面，若您准备好，开始出声即开始计时。
Good afternoon, everyone. I'm Xingqiao Zhou. You can call me Elsa. It's my pleasure to be here and share our product New Plus with you. First of all, I'd like to tell you a story. Look at these beautiful ladies. Well, not me, the other one. Her name is Sonia, and she's my godmother. We are very close. About 2 years ago, she was diagnosed with breast cancer. After 2 surgeries within a month, several rounds of chemotherapy, long-term use of hormone inhibitors, now she's stable. So people tell me that I don't need to worry too much because breast cancer has many drugs, therapies, and an excellent survival. Unfortunately, the scientist in me is more focused on the elevated risk of recurrence and metastasis. Those numbers, 30 to 50% recurrence rate, are a bit scarier. Our products aim to address the unmet medical need of all the Sonias out there who are worried about the 50% to coming back to them in the next decade. The unmet medical need in breast cancers is focused on the challenge of chemotherapy resistance, limited treatment options, high recurrence rate, side effect, and tumor during repair. Our innovative solution is New Plus. New Plus and RNA-based DDB2 inhibitors serve as a chemosensitizers. New Plus focus on the chemotherapy-resistant cancer types. We use computational modeling and stimulation to identify short-screen, high-affinity, low-cost chemically synthesized nucleotide drugs, aiming to address 50% recurrence problem. About our MOA, we found that after chemotherapy, tumor immediately increases the DDB2 level. What is DDB2? DDB2 is damaged DNA binding protein 2. And how NuPlus work? NuPlus prevent the natural DNA repair mechanism in tumor cells, making them unable to repair chemotherapy-induced DNA damages, resulting in increasing chemosensitivity of tumors. About our supporting data show that we found that the NuPlus improves cytotoxicity sensitivity threefold, extend at least 24-hour cytotoxicity effect. At the same time, increase safety by 50% and enhance tumor cytotoxicity by 70%. Up until now, in clinical setting, patients for resistant to chemotherapy, doctors have no better treatment than to repeat treatment. And so the unique advantage of NuPlus, including we could provide a new target biomarker for precision chemotherapies, ER-positive DDB2. And we could provide a wide range of indications and reduce toxicity, increases the effectiveness of existing chemotherapy. About the market share of NuPlus, the serviceable obtained market is 150 million, and the serviceable available market is 307 million. Our acceptable market share for the total addressable market is 13.81 billion across multiple indications, including cholangiocarcinoma, lung cancers, liver cancer, and radiotherapy. About our roadmap. Well, thank you for TGP, Taiwan 萌芽计划 support. In this year, we will finish the GLP validations, and we plan to start our company around 2025 to 26. By 2028, we will finish the GLP validations, and for hopefully coming to the 2030, we can apply the R&D applications. Here is our entire team. Our project leader, co-team member, including the research team and clinical team. We also have several consultants from regulatory, technical, business, and finance sectors provide us perspective and suggestions. NuPlus, that means new treatment option plus an optimized therapeutic effect. We are CMU Professor Wei Chen Huang's lab, and thank you.
非常感谢我们第8组的简报。那我们接下来呢，就要邀请第9组，一样是来自中国医药大学，是詹世轩团队。请您准备好即开始简报。
各位大家好，我是来自中国医药大学的詹世轩，今天为大家介绍的技术名称是治疗性人化 CD24 单株抗体，作为新一代的免疫疗法。那我用这个缩时摄影来为这个专利抗体破题。这个缩时摄影展示的是我们的专利抗体可以诱发巨噬细胞，能够有效吞吃带有绿色荧光的三阴性乳癌细胞，并且在肚子里面快速的消化它。那我们所要聚焦的适应症是转移性三阴性乳癌 TNBC。那 TNBC 呢，它是在乳癌亚型中一个非常恶性的亚型，它的5年存活率不到12个月。那原因是在于病人在手术化疗之后呢，容易产生这个复发跟转移。那临床上呢，它的标靶药物非常少举例来讲 ，PARP inhibitor 大概只能治疗小于10% 的 breast cancer patient。那最后一线的药物免疫检查点治疗啊，它临床效益非常差，它小于18%。那所以在2019年，我们有一个机会我们自己的研究发现的，三阴性乳癌在转移的时候会表现大量的 CD24，然后会去促使癌细胞的生长。那 Stanford Group 它更发现 CD24 跟 CD10 是一个新的免疫检查点疗法。那 CD24 它可以去帮助癌细胞去抵抗来自于巨噬细胞的吞吃，所以有这个机会，我们可以针对 CD24 开发单株抗体。那经过多年努力呢，我们终于开发出一个治疗效果极佳的人化 CD24 抗体，HH0146，那以下简称专利抗体。那我们专利抗体就有以下几个特色：第一个，它可以很好的活化巨噬细胞，让它吞吃癌细胞能力增强10倍。那第二个是它可以增强自然杀手细胞毒杀能力3倍以上。第三个，它也可以促进这个T cell 的肿瘤浸润。那第四个，它可以在小鼠实验呢提升这个小鼠的存活率到达80%。 OK， 那我们药理学实验显示，这个我们专利抗体有非常好的剂量效益，它甚至在每公斤0.5毫克底下就可以抑制95%的tumor 的生长。那我们在另外一个三阴性乳癌的TNBC 的适应症也得到一个证实。那另外专利抗体也显示出它在荷尔蒙阳性跟HER2阳性的这个乳癌 model 也得到一个很有效的抑制。那更重要的是，我们专利抗体可以有效在小鼠的实验可以有效抑制3C 肺癌转移，可以提升小鼠的存活率从25% 到100%。那我们的细胞实验或者是活体实验都可以证实我们的专利抗体呢，可以很好程度的活化巨噬细胞，吞噬癌细胞，而且在肿瘤里面也看到大量的巨噬细胞的浸润。那我们也跟 CDMO 合作，初步探索前期制程。那我们发现我们的抗体呢，是符合这个工业等级量产的一个制程。我们可以在初步的时候可以达到每公升有4.78 gram 的一个水准。那因为我们这个抗体呢是市场的首见药，那我们竞争对手主要有4个，其中2个已经进入 Phase 1。另外2个还是在 preclinical， 那我们也是在 preclinical stage。那我们的技术优势在于，我们是具有专利保护的这个序列新颖性。那我们在我们在这个最佳剂量的时候可以引发肿瘤完全反应，而且我们可以抑制转移疾病，而且我们在0.1 mpk 的这个剂量就可以达到肿瘤抑制。那市场规模分析呢，在可触及三阴性乳癌大分子药的这个市场呢，我们每年大概会有3000万美元。那在主要市场，美国大概会有30亿美元，在全世界的市场大概有100亿美元。那 CD24 在其他的 solid tumor 也有表现增加，所以说如果增加适应症的状况下呢，它市场规模可以达到200亿美元。Okay， 那我们期望我们的专利抗体可以为病人提升存活率，然后带来更有效的治疗，减少疾病的副作用，还有可以增加病人的适用性。那我们的专利 portfolio 呢，我们已经快速的拿到了台湾专利、发明专利。那我们也得到了国际检所的报告是非常的优异。未来我们的计划，我们规划在萌芽计划支持下，感谢萌芽计划，我们已经完成了多个POC 的 study， 那剂量探索还有毒理等相关实验。那未来1年呢，我们需要1500万的一个资金去进行这个细胞库的建制。那以利后续的这个 GLP study， 像是动物毒理还有ADA 免疫原性的治疗，免疫原性的探索。那以完备 IND 前的一个准备。那这个部分的话，我们预计我们的评估的资金还需要1亿5千万。那以下这个是我们的团队成员列表。那以上是我的简报，谢谢各位。
非常谢谢来自第9组的简报，谢谢您，请入席。接下来我们要欢迎第10组，也是今天新药组决选最后一组，来自高雄长庚纪念医院以及中国医药大学林杰兴及蓝仙元团队，为我们接下来进行简报。若您准备好，开始出声及开始计时。
各位评审辛苦了。今天的最后一组报告申请人是我本人以及中国医药大学蓝仙元所长。我们的题目是用 DAAO inhibitor 来治疗忧郁症。忧郁症是一个很复杂而且病因很多的一个疾病，它很容易造成失能。目前的抗忧郁剂呢，很多病人的治疗反应并不佳，特别是在认知功能以及对压力的感知这方面，而且还有很多副作用，包括代谢方面的副作用。那我们发现 NMDA receptor 呢，跟这个忧郁症的病因是有关的。Sodium benzoate 它可以透过 DAAO 的这个抑制来促进 NMDA 的神经传导。所以我们的这个技术是全球第一个以 Sodium benzoate 很安全的，可以去改善病人的忧郁、压力感知以及认知功能。我们在一个117位的一个临床试验当中，我们去比较了 Sodium benzoate 传统的抗忧郁剂 sertraline 以及安慰剂这3组，我们发现经过8周的治疗之后 ，Sodium benzoate 确实可以去显著的改善病人对压力的感知。更重要的是在认知功能的进步，因为我们知道在高龄化社会，罹患忧郁症的病人越来越多。那这个老人如果罹患忧郁症的话，会去增加他这个失智症的风险。所以未来的忧郁症的治疗重点可能会在认知功能的改善。我们发现，在8周治疗之后 ，Sodium benzoate 可以很显著的优于其他2组，去改善病人的认知功能。这个是其他抗忧郁剂没有的特色。好，我们也发现 Sodium benzoate 它病人的这个留置率最高，也就是最不容易 drop out。 另外，我们发现传统抗忧郁剂它会去上升 LDL， 是一种比较不好的血脂肪。那 sodium benzoate 也没有这个问题。好，跟其他的抗忧郁剂比起来，我们有一些优势。目前最常用的是 SSRI 跟 SNRI。那这些抗忧郁剂很多病人的治疗反应并不好，有明显的这个副作用。那而且他们对这个认知功能以及压力感知这部分没有什么特别的疗效。那 sodium benzoate 比起来有在认知功能及压力感知这些优势。那还有一类是这个 MT1、MT2 的 agonist， 但是他们对老人忧郁并没有疗效。那 sodium benzoate 我们有试过在老人的这个忧郁症，我们也可以看到显著的有改善。还有这个 NMDA receptor antagonist， 包括 ketamine 这类的药物。那这些药物呢，它会损害认知功能，而且有明显的成瘾的问题。sodium benzoate 也没有这样的问题。那还有一类就是比较psychedelic 的 drug， 譬如说MDMA 就是像摇头丸啊这类的这些 CNS stimulant。那这一类的药物的副作用就很多了，包括认知功能啊 addiction， 还有很多的其他的不良的副作用。那 sodium benzoate 也没有这些问题。那我们知道，忧郁症是一个盛行率很高的。那根据预估，2030年的时候可能会是排行首位的最容易造成 disable 的疾病。那这个全球的 market size 非常的大哈。那预估到2027年的时候，可能会有180几亿美元的这样的 market size。在全台湾呢，15岁以上大概有9%左右的病人罹患不同程度的这个忧郁，而且这个市场还在成长当中哦。因为有很多的病人，因为不敢去就医啊，或者是治疗的效果不够好，还没有获得适当治疗，所以我们相信这个市场还在大量的成长当中。好，那我们的这个技术目前已经有获得至少6个国家的专利，包括台湾、澳洲、日本、加拿大、马来西亚以及美国，还有其他几个国家的专利目前还在申请当中。那我们目前也在跟一个科技制药公司有在商谈一些技转相关的一些步骤。那如果成功技转的话，我们相信大概在获得 approval 的前5年，大概就可以有大概10几亿台币，以及在海外的话，大概有60几亿美元这样子的market potential。那我今天报告很迅速，那非常感谢各位委员的聆听，希望各位委员多多给予指教，谢谢。
非常感谢我们最后一组的简报，谢谢您，请入席。那么接下来邀请评审移步到我们评选的会议室进行我们的决选。不过在这之前，我们有 Q&A 的环节不好意思耽误了大家来让大家紧张了一下哦。那我们这边呢就邀请呢我们6到10组的决选的参赛团队代表来到台前，一样请您面对评审由左至右按照编号入席，请派一位代表入席即可。那在大家入席的期间呢，不耽误大家太多时间，我快速的再次的讲解这一次 Q&A 的流程。25分钟的总 Q&A 时间，采评审一问一答，每一题的答题时间只有2分钟，1分钟会按铃1次提醒，2分钟会按铃2次，请立刻停止简报。那若评审准备好，我们开始 Q&A 环节，谢谢。
第7组哈，因为它是靶向血小板的 fusion protein， 我了解就是非一种蛇毒蛋白嘛。
是。
那这个 fusion protein 是一个 foreign 的 protein， 那 what's the potential for generating neutralizing antibody？ 如果有 neutralizing antibody， 就等于说我用了几次之后就会没效。
是，谢谢委员的提问。那针对这一部分，我们事实上有做过所谓 neutralization 的 antibody 的一个 titer， 还有它的一个测试。那我们早期的资讯里面显示，就是说我们的 neutralization antibody 的不会针对我们的就是 active site 那边有辨识，那这个算是我们欣慰的地方。那另外一个进一步就是说，我们想要接下来的改良，就是把它改良成比较小的一个部分，那就是我们之后的一个方向。那避免所谓的 antibody 的 effect。那另外有提到就是说，我们早期的 animal model 都是在针对单体的部分，就是说蛇毒蛋白的部分。那后面会用到 fusion protein， 就是跟 TPA 的2代药就是 TNK 做 fusion。那这个一部分也是为了要所谓降低它的免疫原性的一个问题。对。 那我们事实上也有做另外一个所谓的一个 half life， 也就是它免疫原性的改良，就是跟 FC 或是一个 HSA 的 fusion protein 来做一个改良。那这部分就是我们针对这一块的一个设计，在未来之后都会有一个这方面的一个所谓的改良药物的出现。好，谢谢。
我这边有一个问题啊，想问那个万能抗体所，我想了解一下，就是说你这个是用那个 peptide block 的 antibody variation region， 然后就是说我想知道，就是说你这个是one size fit all， 就是你只要有一个抗体所，你就可以就是说 apply to all different cancers。
谢谢委员的提问。就是我们的抗体所主要是利用抗体腰部 hinge 的部分，然后我们利用基因工程的方式，让它加在抗体的轻链跟重链的 N 端。那透过这样子的 disulfide bond 的话，它就可以形成一个空间性的屏蔽。那这样子的抗体所，它可以 one for all for 任何的抗体，不像我们竞争者，他们是利用亲和力的屏蔽，利用 bacteria display 的 library， 他们筛选一段亲和力的胚胎，黏住抗体的 binding site。之后再去做竞争，那这样子的话，我们的抗体就可以快速的应用在任何的抗体上面。但是我们会针对不同抗体所针对的适应症，利用我们的肿瘤简体库去筛选出来，面对这个疾病，我们要使用哪一种的 protease substrate 当做 linker， 让它有更好的解锁效率。所以在 protease linker 这边的话，有可能会针对不同的疾病，我们会去做不同的设计。
所以不同的癌症，你们用的那个锁就不一样，因为因为不同的这个 cancer 的 in terms of the level of protease will be different, right? So 有些cancer will be higher level with a certain proteases, right? So pretty much a different cancer you need to use different peptide lock.
对。就是在手的假装我的手是 linker 的话，这边是我们的抗体所的话，我的手会有多种的设计。那我们其实现在目前有15种的 linker， 然后它可以去做设计，然后我们还会利用电脑的方式去做突变。那一个部分的话，也是为什么我们可以让每一个抗体都有每一个 lock antibody 都有新专利的 know how。
所以你们是 screen for like 500 samples, try to identify which peptide, which protease is higher in a certain cancer type. 所以你们现在是发现，就是说，是不是不同的癌种，它的那个protease 也不一样 in terms of expression level, different proteases and different expression level. 所以你们针对这个去设计你们的抗体所。
这个问题是我们现在目前竞争者他们还没有解决的部分，但是面对这个问题，我们已经有找到一段 substrate， 它是像我们 EGFR， 它是可以用在大肠癌、头颈癌跟肺癌。那我们这一段 substrate， 其实在这3种癌症上面都有80-90% 的解锁效率，我们才选用了这一段 linker 去做后段的开发。
OK，
好，谢谢委员。
我想我有个问题想请 ，Well, maybe I should speak in English, because you spoke in English. So I think that you mentioned about the using the approach of nucleic acid as the chemosensitizer.
Yes.
I'm wondering what is your clinical strategy, because there are so many chemo out there, and then many of them are in combo. So I think that your speed to your clinic and complete your clinical trial will be quite hinged upon how do you approach this from the clinical trial design point of view. I wonder whether you can elaborate that. Thank you.
Thank you for your asking so important questions. And we do discuss with our clinical trial for the Dr. Huang, and in our clinical trial design, before that we try to enter the neoadjuvant, but we found out that the real unmet medical need, especially is chemotherapy resistant patients. So there will be the second line. That means that the after treatment of the chemotherapy later, if resistant, then new plus could add on with the like the lipo dogs, because lipo dogs that people usually pay for, and in the especially for the meta breast patients, is the 100% will use the chemotherapy, and they used to for the maximum dose to pre-treat or retreat again. So our TA is focused on the second line for the chemotherapy-resistant patients.
那个第9对啊，就是 CD24 的，是 CD24 是一个 new target 哈，它是有一点点的热度，因为它是一个 immuno checkpoint for meta 跟这个 invasion 哈。所以它可能的效果 in cytotoxic 不是那么强。那不晓得你的 animal data 是显示出来，它是有就是杀癌细胞或者是让癌细胞不长的这个效用。那你的 preclinical data 跟这2个 phase 1的 compound 比较是如何？
OK， 谢谢委员的提问。那 CD24 我们一开始是发现它在转移的时候才会表达，那所以我们认为它在转移性疾病是很有希望的。那我们在做这个 CDX model 治疗的时候，它的疗效是非常好的，它的剂量0.5 MPK 它就可以抑制掉肿瘤生长但我们也提到，就是委员提到就是它的毒性、它的安全性的问题。那我们在小鼠的可比就是鼠类的替代抗体的一个小鼠实验。那我们也看到它的剂量在5 MPK 的时候呢，它是对身体的器官没有什么样很强烈的明显毒性反应，但这个部分我们还是继续会做探索。对，那目前为止就是我们这个抗体的部分呢，我们在未来就是想要看，就是在委员刚才提问就是 competitor 的部分，那因为他们这个是市场是首见药，所以目前为止我们拿不到他们的抗体，但是我们已经跟公司合作，可以去把他们专利抗体的序列调出来。那我们现在已经初步核准他们的抗体，那我们现在下一步正要进行 head to head 比较，看看我们的疗效好还是他的疗效好。
对不起啊，追问一个问题。因为 CD24 有一个 small protein core with lot of glycosylation， 所以 in different cell 可能 glycosylation 这个遮蔽的 degree 都是不同的，那就不晓得它的 diversity 怎么样。那你用一个 compound 可以 universal 抑制吗
？One antibody for all cancer。目前为止，我们知道 CD24 的 glycosylation 在 depends on cell type 或者是 cancer type 会很不一样，所以我们那时候在screening 这个 antibody 的时候，我们 screen 非常多的breast cancer cell、hormone positive、HER2 positive 跟 triple negative， 那甚至 liver cancer 还有 pancreatic cancer， 我们的专利抗体都可以辨认到它细胞膜上面的CD24。那所以我们去进行了这个 epitope 的一个 exploring， 就发现说它其实是可以辨认到它的 peptide core， 所以它有可能是可以规避掉它的这个 N-glycosylation 的部分，所以这部分我们有做一系列的调查。
我想请教一下这个硕准这个团队，就说你的这个 luck 啊，会不会影响原来药物的这个 PK/PD？ 那原来药物它已经就是做完所有的 clinical trial， 它的 PK/PD 其实 well characterized， 但是你去改变了它的这个东西，那会不会影响它原来 PK/PD？ 第二个是你的 luck pretty much rely on 这个 tumor microenvironment， 它的这个 protease， 可是我们很难去 characterize， 就是说这个不同的病人他的 TME 的这个 protease 的这个 expression， 它的 level， 它的这个 specificity， 所以这个会不会造成了你在药物开发的另外一个 uncertainty？ 谢谢。
好，谢谢委员的提问。那针对第一个问题，有关于 PK/PD 的部分，如果是 PSN101 针对 anti-EGFR 的这个抗原标靶的话，我们在小鼠上面是发现 PK 不会有任何的影响，但是如果当我们把这个抗体所加在譬如说 mouse CD3 的 antibody 上面，这个在小鼠上面很多抗原表现的抗体的时候，我们发现我们的抗体所反而可以prolong 我们的半衰期，我们可以避免 antigen sink effect 的现象。那这个特性的话，也是我们抗体所的另外一个优势。 那第二个的话，有关于说就是肿瘤抑制性的问题，我们现在就是发现我们其他的竞争者，他们一开始的时候就是要怎么去解锁，大家都只关注怎么锁，但是解锁的时候，通常我们之前就在学研团队的时候，就只有看到譬如说 NMP29， 它就是有过度表现，那我们就选用这一段 link 去来解锁。但我们后来的时候，就是有选择超过150个肿瘤检体，然后我们去利用冷冻检体去筛选出来说，利用蛋白酶的 prop， 我们就可以知道说到底哪一些 protease 它的 link 可以去解锁的效率比较好，去解决像委员刚刚提到的这一个担忧的部分。对，好，谢谢委员。
我有问题要问蓝医师啊，有关于 NMDA， 你刚刚的报告有提到这个一些 data， 事实上已经用在 comparator， 还有用在病人身上了。那但是我们的了解是，这个 sodium benzoate 是很旧的东西。那有提到有一些专利保护，那你的 IP 到底布局是怎么样？能不能说明一下？
谢谢老师的提问。它是一个很老的 compound， 那其实它已经是有个药证的哈，是对一个罕见疾病，所以这是一个新的适应症。那跟旧的来说，它其实有很大的差异。旧的那个剂量非常大，我们这个相对剂量很少。那刚刚老师提到，我们这个已经有那个6国的专利的保护了哈，台湾、澳洲、日本、加拿大、马来西亚跟美国。那国际专利其他的也包含那个中国、欧洲也在审核当中，所以我们有非常好的专利保护。那当然就是说，那将来他会不会说他就是off-label use 那也没问题，因为我们这个是我们正在跟这个原生产的这个公司科进制药股份正在洽谈技转，也就是说，如果他们要 off-label use 的话，还是这个获利还是回到这个原来的公司，所以我想这个就是获利的方面应该也没有什么特别的问题。以上报告。
所以您的 IP 布局事实上是一个 new indication 嘛，是不是？
是 new indication， 是就是针对预诊的，是，谢谢。
那我想接着问，下来问就问蓝老师啊，因为我看了一下那个 clinical.gov， 你们其实临床已经在2020年就做完了嘛，还是有新的，也许有新的这个 trial。我想我的这个 interest 是说，就是说你们现在等待下一个目标是要在临床继续推进呢，还是说一定要再找 partner？
跟委员报告，就是我目前在已经在跟这个科进制药股份有限公司正在洽谈技转了。那技转之后，他们当然就是可以来进行这个上市的 clinical trial， 因为我们这个原先这个是 investigator initiated 的 clinical trial。大概是这样，像老师报，当然就是我们已经非常 solid 的，就是有2个 clinical trial， 那大概都是可以验证。我们相信未来的上市的这 marketing clinical trial 应该可以相当顺利来进展。谢谢。
那另外，我可以继续问哈，我问那个就是 Universal Block， 我想请问，就是说，因为你们这个 adding additional 这个，你刚刚说用手嘛，对不对？从在一般制造的观点来上，你们有没有碰到什么有关于 CMC 上面的问题？比方说 stability 也好啦，或者是说它的 expression level 也好。那第二个问题，我知道我只能问一个问题，那你可以分2次回答，没关系。那第二个问题就是说你的 dose， 就是说 when you compare with the， 就是说原来的这个，就所谓的原来的这个 biologic， 你刚刚讲的是 Erbitux，right？ 就是说。How would you project your dose？ 就是说，因为你的 dose 如果说比它要来得更高的话，也许你要 control 你的 cost of goods。就是你们有没有想到这个所谓的 cost of goods？ 也许有点遥远，只是想听听看你们的想法。谢谢。
好，谢谢委员的提问。那针对第一个问题的话，就是等一下，我先是回答第2个部分，就是 cost of goods 的话，目前的话我们是。近期已经打算要做 tissue cross reactivity， 那我们就可以针对说，我们的 PSN101有不同的剂量，然后它去评 相比我们现在目前的 current treatment 的 Orbitas， 那我们的剂量是可以拉到多少的时候，它是有相似的 binding ability， 那我们就可以也知道说我们的抗体所可以拉到多少的剂量。那我们之前的话，在 CDX 的 model 的话，则是有发现说，原本的 Orbitas 它只能用在10 mg per kilogram 的 dosage， 但是我们的药物借由抗体所的话，它其实是可以拉到25 mg per kilogram 的 dosage， 然后把肿瘤压制得更加的好。对，那第一个问题的话，不好意思，可以麻烦委员。
就是有关于你们在生产制造
生产制造的部分，就是 PSN101的话，目前我们已经完成稳定细胞株的建构。那上游的话，作为 antibody engineering 的 antibody 的话，我们是可以接近4克 per liter。那现在目前已经在进行5公升的抗体量产。那后续的话，我们会请 CDMO 厂帮我们进行 MCB 以及50粒的量产。那目前的话，我们跟其他的双功能抗体去做比较的话，我们认为是一个还算符合产业化规格的技术。对，谢谢。
我想请教一下庄伟哲庄老师这一组团队，你提到就是说你的 molecule 在这个17个小时之后的 IM 还有50% 的 activity。那我 assume 这一类病人他在 disease onset 可能24小时之后，他不是在 ICU， 都还是在这个 inpatient 的这个 care 里面。那你们 claim 就是认为说17小时 IM 还有50% 的这个 activity， 那你们觉得它的 potential application 可能是哪一些地方？谢谢。
跟委员报告一下，这边就是我们事实上我们的蛋白的优势，事实上是我们在测试之后，我们觉得我们可以让所谓的 integrin 在血小板上面的 integrin alpha 多巴胺，它可以保持在一个 resting state。那所以，假如我们是期待是说在不管是在救护车或者在一般的我们没办法做一个紧急治疗的情况底下，我们施打我们的药物，第一个高安全性，那我们的 half life 又长的情况底下，我们期望是可以让所谓的血小板都维持在一个 resting state。那不会有造成进一步的所谓的血栓的问题。那所以我们这一块在half life 的这一块，我们觉得这个时间是我们觉得算是一个很不错的一个因为它跟血液施打的一个方式来比的话，它事实上是蛮一样的，蛮一致的。所以我们会觉得它有个优势是在这一块的治疗都是一个很不错的一个维持在 resting state 的一个不错的治疗上。
让我有个 follow up 问题，也是问我们成大的团队哈，那老师这个 unmet medical need 的确是存在的。如果我们再想的远一点，要有 commercialization 和 BD 的可能的话，你们有没有想过，哪些公司最有可能买你们的技术？
谢谢。
为什么？
谢谢委员。我们有涉及到国内的话，其实也有想要开发的就是一个安全性的药物。那我应该可以直接讲，就是顺药的部分。顺药部分它事实上有，近期它也有进入2B 的 clinical trial。那它其实是一个 peptide 的部分，那它是跟我们第二代药物有点像，它是帮助所谓的 TPA 的药物，可以让它进行一个比较不会有所谓的side effect，就是乱切，然后会有出血的问题。那所以其实我会觉得我们也在测试我们的跟 TNK 就是 TPA 的二代药物的 fusion protein 上，我们的确在 therapeutic Windows 那边也有比较好的一个效果。那我觉得这是一个国内的一个厂商可能会有兴趣。那另外国外的，事实上他们也是做一个改良的，那他们也是进入到 Phase 2的一个 clinical trial。那我觉得这2间公司就是未来，我觉得他们应该都有兴趣针对我们这一块做技转的可能，因为我们的 target 跟他们是不一样，他们都是针对 TNK 做安全性的改良，但是我们是针对血小板的。那这一块是我们一直都认为是说血小板是一个另外一个它算是一个并行的一个治疗的一个很好的 target。一个方面是可以减缓所谓的再复发的一个问题。那我们这一块也可以针对就是这一块有提供高安全性的保护这样子。 谢谢委员。
继续问第7组。我有注意到你有做 IM， 因为一般来讲，像这种药都是 IV， 对，不晓得为什么要做 IM。然后 IM 有显示出来，就是你的 half life 是 dose dependent， 也就是说在低剂量的时候，它的 half life 非常短。请问原因是什么？
跟委员报告一下，就是说，我们目前这个 IM 的部分是测试单体的部分，那它的分子量是小于10 kilodalton 的，那所以我们初步认为，就低剂量的部分的话，应该就是清除的会比较快一点。那所以这一块它的 half life 会相对于短一点。那可是我们有去发现，就是说它的 half life 相较于治疗效果上，我们觉得也是蛮不错了。也就是说，它的达到安全性相较于说它这一块已经算是不错，因为我们可以提高到高剂量的情况底下，它也不会有出血的副作用，所以我们其实就可以只用高剂量的方式去治疗，那一样可以让它长时间保持在血小板是一个 resting state 这样子。谢谢委员。
我想请教一下那个 CD24 抗体的开发。那 CD24 也类似就是Don't Eat Me Signal 的一个成员之一。那 Don't Eat Me Signal actually there's quite a few Don't Eat Me Signal. Now 也都是 the focus of the drug discovery. 那你现在就说以 CD24 当做 target， 你怎么跟就说 what's the benefit？ 为什么你会特别就选这个 CD24 as your target to develop the drug as opposed to other don't eat meat signal？
回答委员的问题，那是人类历史第一个don't eat meat signal 被发现是 CD47。那他最近去年他 fail 在 clinical phase 2， 那原因是因为他有RBC cell 上有大量表现，所以说第一个 don't eat meat signal 已经 already fail。那所以我们在自己的开发过程，因为我们当初是 focus on metastatic triple negative 这个 clinical unmet need， 所以我们当初其实没有预期我们会找到这个分子，那是因为它在转移之后细胞分泌出来表现大量 CD24 才被我们找出来说原来这个 CD24 它是在转移的时候会表现，那会增加这个转移的致命性。那由于metastatic triple negative breast cancer 它在临床上几乎是没有药医的，所以说这样子的一个 target 就变成是triple negative breast cancer 目前为止是一个很好的一个therapeutic target， 所以我们开发这个适应症的方向。
所以 CD24 是 specific to metastatic triple negative cancer, or actually is applied to all other metastatic cancers?
事实上，在2015年的一篇 Cancer Cell， 它有发表在 prostate metastasis to lung tissue， 它也有侦测到很高量的 CD24 的表现。所以我相信不是只有在 triple negative breast cancer。那我们现在所知的科学上的证据显示，就是至少现在目前观察到prostate cancer metastatic disease 或者是lung cancer metastatic disease， 再加上 triple negative cancer metastatic disease， 它都有表现高量的 CD24。那后来我们的机转研究也发现 ，CD24 这分子很特别，因为癌细胞要转移到远端器官的时候，它必须要从 mesenchymal type reverse 到 epithelial type。那它在这中间过程呢 ，MET 这个 phenotype transition 扮演很重要的角色，所以有可能这是为什么它在 metastatic disease 在不同的 cancer type 它都有大量表现。
好的，谢谢评审提问，也谢谢我们的决选团队的回答。那我们呢决选 Q&A 时间目前已经结束，再次的感谢大家，我们请决选团队先入席。同时再次感谢评审以及今天所有参与决选的团队，以及在现场我们一起聆听今天决选作品的大家。我们现在呢要邀请评审团队，现在先移步至我们的评选会议室进行会后的讨论。谢谢您，我们掌声送给评审。 今天的决赛结果会在明天闭幕式时为大家揭晓。那呢，这边也要和大家预告，明天我们的报道时间是9点钟，一样为大家安排了专题演讲以及趋势论坛，要跟大家探讨呢创新生医的相关的问题。那同时在下午的时候呢，我们还会有呢第2场的决选，也就是医材组的决赛，所以再请各位来宾们不要错过。明天的报道时间是9点，议程开始的时间是9点半，再请各位呢帮我注意时间。那目前呢，现在台风的天候状况稍微比较严峻，明天请各位来宾前来的时候，以及今天离场时都务必注意回家路上的安全。谢谢大家的参与，我们明天再见。